the national anthem of the Republic of Indonesia, Indonesia Raya. Thank you. Please be seated. Assalamualaikum warahmatullahi wabarakatuh.
Good morning, Your Excellency, the Chairperson of the Indonesian FDA, Professor Taruna Ikrar, MD, and Biomed, PhD, Indonesian FDA colleague, distinguished guests, respected speaker, ladies and gentlemen. On behalf of the organizing committee, it is our great pleasure to welcome you to
The International Seminar, Perspective from Five Continents, the Laboratory Future, Sustainability in Innovation and Operations. Ladies and gentlemen, today we gather not only to discuss scientific advancement, but also to share a collective visions for the laboratories of the future. In an era characterized by rapid technological transformation and increasing global challenges, laboratories are expected to be innovative, digitally connected, environmentally responsible, and internationally collaborative.
This seminar provides an important platform from regulators, scientists, academia, industry representatives, and laboratory professionals to exchange ideas, strengthen partnership, and explore innovative solutions for sustainability laboratory operation. May today's discussion inspire meaningful collaboration and contribute to the advancement of laboratory excellency for the benefit of public health.
Before we begin this seminar, please allow us to introduce ourselves. My name is Nia Yuniarti, and together with my colleague,
Saras,
we have the honor and privilege of serving you as a master of ceremony of today's international seminar.
we are delighted to accompany you throughout today's program and will do our best to ensure that the event proceeds smoothly and enjoyably. Once again, welcome. Thank you for joining us. We hope you find today's seminar both insightful and inspiring. Distinguished guests, ladies and gentlemen, today's seminar reflects our shared commitment to strengthening laboratory excellency, fostering innovation, and advancing sustainability in support of public health. Before we proceeding the opening remark, we shall first hear the activity report regarding the organization and objective of this international seminar.
It is now our pleasure to invite the head of Center for National Quality Control Laboratory of Drug and Food to deliver the activity report. May we respectfully invite Ibu Mimin Jiwawinanti, SSC Apoteker. The floor is yours.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera bagi kita semua. Good morning, Distinguished Chairperson of Indonesian FDA, Profesor Dr. Taruna Ikrar, speakers, Dr. Tindo from SPTLC Association Switzerland, Mr. Chris Chia from United States of America, Dr. Sheikh Umar from WHO Regional Africa, Mr. Blad Clark, University of Melbourne, Mr. Sudip Sabdi from USP, Dr. Elvis Okovo from University of Queensland, Dr. Saturnan Kartikeyan from Frontier Laboratory Singapore, Mr. Aaron Desmond from Green Laboratory USA, pimpinan tinggi media, staf ahli, pakar ahli, pimpinan tinggi Pratama, PFM ahli utama, representative from Ministry and Government, representative from University, representative from Indonesian Manufacturer, representative from Badan POM Laboratory Network, Indonesian FDA colleagues, and all participants. May I respectfully report to Distinguished Chairperson of Indonesian FDA, Profesor Dr. Taruna Ikrar, regarding the implementation of the international seminar entitled Perspective from 5 Continents, the Laboratory Future, Sustainability in Innovation and Operation. This seminar will be organized on July 6 and 7 to 2026 in hybrid format. The seminar brought together experts, regulators, academia, and also laboratory professional from 5 continents to discuss recent development and future direction in laboratory science. And this seminar was held to strengthen laboratory capacity and promote international collaboration in response to the growing of challenges that we facing in the drug and food laboratory, and also rapid technological advance, the increasing complexity of global supply chain, emerging contaminants, and also the need of environmentally sustainability laboratory operation that we require to continuously innovate while maintaining scientific integrity and compliance with international standards. This event comprised internal BPOM launching activities, the presentation of award to regional offices of BPOM, and also the signing of the cooperation agreement with GP Farmasi. Bapak Ibu sekalian, bahwa pada pembukaan acara ini juga akan disertai oleh aktivitas yaitu launching panduan teknis interaktif berlaboratorium yang baik, penyerahan buku pedoman penjaminan kualitas pengujian laboratorium BPOM, penyerahan penghargaan kepada UPT atas kontribusinya dalam pengawalan UMKM paham uji, dan juga penandatanganan perjanjian kerjasama dengan GV Farmasi terkait pengembangan metode analisis. This activity will further strengthen Badan POM commitment to building laboratory capacity, improving quality assurance system, and fostering collaboration with stakeholders to develop reliable and science-based analytical method. The seminar brought together distinguished speakers from 5 continents representing international organizations, professional association, industry, external laboratories, and also academic. And the technical session cover topic including HPTLC, digital transformation and laboratory information management system, laboratory automation, green laboratory, microplastic, emerging contaminants, rapid microbiology method, and quality assurance and global standard. And the seminar was attended by a representative from Indonesian FDA as well as external participant from laboratory, academic institution, government ministries, industry, association, and other relevant stakeholders. The seminar attended by 250 in on-site participation and also approximately 950 online participation. And the external participation included laboratory network of BPOM, Indonesian Manufacturer Association, GP Farmasi, Perkosmi, GAPMI, GP Jamu, and APSKI, also universities, including University of Indonesia, University Defense University, Indonesia Defense University, Pancasila University, IPB University, Gajah Mada University, Airlangga University, and also University of Jember, and pharmaceutical and food industry. Through presentation, discussion, participant exchange experience, share best practice, and also explore innovative approach to develop adaptive, sustainable, and interconnected laboratory. The seminar enhanced participant understanding in sustainable laboratory operation and emerging technology, while strengthening collaboration among regulators, international organizations, industry, and academia. The outcomes of this event are expected to contribute to development of innovative laboratory system and to support continuous improvement of drug and food control laboratory in Indonesia. Distinguished Chairperson of Indonesian FDA, Professor Dr. Tarina Ikrar, we respectfully seek your guidance as we continue to strengthen and advance our laboratory and also the laboratory network to address the future challenges and also seize emerging opportunities. We kindly request the Chairperson of Indonesian FDA to officially open the international seminar. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you very much for the head of the National Quality Control Laboratory of Drug and Food for comprehensive report and for highlighting the vision and objective of this important international seminar.
Okay, distinguished ladies and gentlemen, as we continue with today's program, we invite you to get to know the chairperson of the Indonesian FDA more closely, a visionary leader who continues to champion innovation, integrity, and institutional transformation in safeguarding the quality, safety, and efficacy of drug and food in Indonesia. Please enjoy the following video.
This journey began in the world of science. For more than 2 decades, Taruna Ikrar has dedicated himself to biomedical sciences through pharmacology, cardiology, and neuroscience. His contributions to functional imaging techniques, as well as cell and gene therapy, have led to breakthroughs in the treatment of rare diseases. His scientific works have been published in internationally renowned journals, including 6 publications in Nature journals. More than 100 scientific publications and several books have been produced, earning 2011 citations on Google Scholar and more than 1,900 Scopus indexed citations. His publications are listed across Q1, Q2, Q3, and Q4 journal categories, with an H index of 19 and an I10 index of 26. In 2022, he was recognized among the top 1% most influential scientists in Indonesia, among more than 6,000 professors, 45,000 doctorate holders, and 250,600 lecturers registered in SINTA. His contributions have also been recognized internationally through the Presidential Award for Outstanding Scientist. Honorary professorships have been conferred upon him by various universities in Indonesia and around the world. His ideas have also reached leading global universities, including Harvard University, Tsinghua University, the University of New South Wales, and the National University of Singapore. His academic journey and contributions to society led him to be entrusted with the leadership of the Indonesian Food and Drug Authority. Today, he continues his dedication to strengthening Indonesia's national food and drug regulatory system. Under his leadership, Indonesia achieved the WHO listed authority status, a global recognition that also earned him the Visionary Leadership Award. Taruna Ikrar continues to devote his knowledge to the country, ensuring that every product consumed by the public is safe, effective, and of high quality. Please welcome the chairperson of the Indonesian Food and Drug Authority, Taruna Ikrar.
Okay, ladies and gentlemen, it is my distinct honor to invite the chairperson of the Indonesian FDA to deliver the opening remark. May I respectfully invite Professor Taruna Ikrar, MD, M. Biomed PhD, the chairperson of the Indonesian FDA. Your Excellency, the floor is yours.
Thank you. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everybody. Honorable International Speaker, Dr. Tindo, Secretary General, HPTLC Association International, Mr. Chris Chick, Application Manager, Chromatography and Mass Spectrophotometer, Thermo Fisher. And also we have it by online, I think, Mr. Aaron Desmond, Green Lab USA. Also Mr. Jack, Market Development Manager, Water in Asian. Sudip Sabde, Senior Scientific Affairs Manager, USP USA. Associate Professor Brad Clark, Australian Laboratory for Emerging Contaminant, the University of Melbourne. And also in online is Mr. Nicholas Famakos, Principal Commercial Manager, Waters Europe. And the last one is Dr. Sheikh Umar Kolibe, WHO APRO. And Chairperson of GP Farmasi Indonesia, Mr. Tirto Kusnadi. Ladies and gentlemen, actually also from here is their Research Center Pharmaceutical Raw Material, Mr. Sofa Fajria, Mrs. And Vice Rector for Planning, Collaboration, Information System, Professor Dr. Bambang, Professor at University of Airlangga, Professor Dr. Ida Kusumawati, and distinguished guest participant and colleagues. Today is actually wonderful day because we saw there's something specific we will talking about. We're talking about laboratory and innovation. Also, we're talking about sustainable and interconnected between continent, and we speak about 5 continents. But let me add something that actually we have it something very important regarding the laboratory. Because whatever we do so far, basic or based on of the scientific results should be regarding the good laboratory. Without good laboratory, how we can get good result? Of course, sometimes we saw, sometimes the laboratory test result is positive, sometimes is negative. But because the technology is developed very well, that's why there are specific scale or specific parameter we have it to use it. And the Indonesian P3OM, our laboratory, I believe we already have it about 100 units from Europe in around our country. But we don't speak only about the number. We speak about the quality. That's why today's meeting is important. Today's conference is important because we believe that if we can develop more technology, and this developed technology can be sustainable and also is innovation technology, and we know also not every laboratory have strength. Always they are the weak side. That's why we need interconnecting between one laboratory to another laboratory. What the purpose for that? Of course, we needed something interconnected because the challenge. In our era, we saw that challenge is wonderful we need to face. For example, so many things, even drug or food, will contaminate for our environment because our environment already changed. We understand that microplastic, even nanoplastic already impact to our environment. Based on of the one reference for my opinion about this is just published in New England Journal in 2021. The discovery is they saw there are stroke, there are atherosclerosis in the brain and triggered by microplastic. So far we understand their blood-brain barrier in our brain. But the problem is how the microplastic can go in through to our vascular system. We don't know yet, but maybe because they are nanoparticle from this microplastic and going through to our blood and stuck in some and it make injure in our endothelial system inside our vascular system. That means can create injury and can create like ischemic in our brain, and finally they make stroke. That means there are something very important in our body. They protect very well, but why the microplastic can go in through to our brain? This, in fact, because the environment already changed. We use it a lot of plastic, and the plastic is not prepared well. The plastic can harm us. So, I just very wonderful discussion with the Parliament about last month about this issue, that we want to make sure there are specific scale to measure this parameter, to scale international scale about how and how much plastic or nanoparticle or microplastic can going through and become safety, be drink or be used by or consumed by our people. The international standard, we don't have it yet about the microplastic. Only one thing about this, that's how important laboratory. We don't speak yet about, we say the drug, especially for narcotic particle. We understand that the protein for the illegal and also so many cases in our community, they use something illegal medicine, illegal drug. And also to develop this one, we need parameter, and parameter is correct, should be tested in laboratory. That means we have a lot of things around the world, not only in Indonesia, in any continent, we need specific tools, specific facility, specific laboratory to test many things in the phase. We need to change this and to help our people. Another thing, we understand that as a regulatory, the Indonesian Food Drug Authority, or BPOM, we have three very important duties. First, we make sure every product we use it, we consume, or our people consume, should be safety. The second thing, we need to also confirm every product is going through to our people should be a good quality. And the last one should be good efficacy regarding the claim, for example, the drug regarding the clinical trial. But from this side, safety, quality, and efficacy, the important thing is to protect the people. Then, to do our duty, we must do something by scientific result. And scientific results should be going through to our laboratory tests. That's why laboratory test is very important in any regulatory, not only in regulatory government laboratory, but also in university laboratory also need a specific scale and need good quality as well. And also in industry. So for this reason, that's why we need to interconnect it between one laboratory to another laboratory. As a chairperson of Indonesian FDA, I'm happy because the Indonesian FDA laboratory already preparing for the green laboratory. And so far, this program already is 1 year, and we will move forward to keep it, this program, to make our laboratory system, our laboratory parameter, and our laboratory result should be regarding the high standard. So, for that reason, I believe we need interconnected and collaboration to many countries, not only with Indonesian. So far, in the Asian, I think we already have commitment. We say the mutual recognition agreement between Asian countries. But this should be more, not only in Asian country, but we need more to other countries in the other continent. That's why it should be the international relationship. We make the strengthening relationship because not only for Indonesian alone, but for everybody around the world. And I believe by developing one system, one single relationship between around the world, and we increase our integrity, I believe the patient safety, the people can be use and consume any product, even drug, cosmetic, or even the food, should be safety, good quality, and efficacy. And I believe by today's meeting, a lot of ideas can develop in this room, and by developing that in the future will be a new innovative idea can be available in all of us, in our community, include in Indonesian FDA or include in the Badan POM lab. And the last one, I want to also mention about something very important about the develop or science and technology, specifically belonging to the digital transformation and implementation of laboratory information management system. Indonesian FDA currently develop the artificial connecting system with apply for our relationship to make every product want to register in our country registered to BPOM should be there connected to our laboratory, specific accredited by our accredited institution. Another thing about one important thing about the robotic and laboratory automatic system, we do understand too that a lot of tools currently available. And to make modernize our technology in Indonesian FDA, we collaborate with other institutions, just for example, WHO, and also we do also to preparing the funding for that. We already also collaborate the Asian Development Bank to prepare our laboratory be ready with a lot of challenge in the future. And the last one, I want to say that the Indonesian FDA already recognized that strengthening laboratorium capacity is essential to protect public health and supporting our economy development. Like we tell you before, that Indonesia is really crucial country because we are one of the four very top big population, and also we have the place is very important position because the Indonesian non-ally country means we will have a friend whatever in the North America. We will have friend with WTO, we have friend with the BRICS, so we are not allied with just one country too. But Indonesia is independent country and crucial important country because we have is high ambition to be the higher income country. So I believe in the near future, Indonesia can be one of the top or 5 top, the biggest, not only the biggest population, but we will the biggest economy and important economy around the world. And the last, but it's not least, is I wish this seminar will inspire us. Is a new idea, strengthening international partnership and contribute is a meaningful to develop all of us. And always I think collaboration is better than competition. If we do collaboration, everyone will win. But we have competition, one win, but other will lose. What we don't want to be lose, we want to be winner, and the best winner is if we winner together. That's why the keyword collaboration is better than competition. I wish this seminar can good benefit for all of us, and by announce say Bismillahirrahmanirrahim, I open this seminar. Thank you very much for your attention.
Thank you very much, Prof. Taruna Ikrar, for the inspiring remark and visionary insight. Your Excellency, Prof. Taruna Ikrar, may we kindly invite you to remain on the stage as we continue with the next part of today's agenda.
Ladies and gentlemen, we shall now proceed the ceremonial session, which marks several important milestones in strengthening laboratory excellency, quality assurance, and strategic collaboration.
The first ceremony is the launching of the first series of the Interactive Technical Guideline on Good Laboratory Practices atau Panduan Teknis Interaktif Berlaboratorium yang Baik seri pertama, and Guidelines for Quality Assurance of Laboratory Testing Results of the Indonesian FDA atau Pedoman Penjaminan Kualitas Hasil Pengujian Laboratorium Badan POM. To proceed this agenda, may we respectfully invite the Honorable Permanent Secretary, all deputies of the Indonesian FDA, the Principal Inspector of Indonesian FDA, and the head of Center for National Quality Control Laboratory of Drug and Food. And also we invite the Honorable Dr. Rita Endang Apotheker MKes to come forward on the stage.
Okay, distinguished guests, ladies and gentlemen, we are now honored to present the official launch of the first series of interactive technical guideline on good laboratory practices atau panduan teknis interaktif berlaboratorium yang baik seri pertama and guideline for quality assurance of laboratory testing result of the Indonesian FDA atau pedoman penjaminan kualitas hasil pengujian laboratorium Badan POM. May we respectfully invite the chairperson of the Indonesian FDA, Professor Taruna Ikrar, MD and Biomed PhD, to officially launch by touching the digital launch panel. Ladies and gentlemen, let us count down together. 3, 2, 1.
Badan POM mempersembahkan dua inovasi untuk memperkuat mutu hasil pengujian. Pedoman penjaminan kualitas hasil pengujian sediaan farmasi dan pangan olahan di laboratorium Badan POM. Sebuah acuan untuk menjamin kualitas hasil pengujian secara konsisten dan terstandar, memenuhi prinsip validitas sebagai dasar ilmiah dalam pengambilan keputusan pengawasan. Badan POM juga menghadirkan video interaktif panduan teknis berlaboratorium yang baik. Program ini dirancang dalam 4 seri yang saling terintegrasi untuk mendukung pengembangan kapasitas laboratorium secara berkelanjutan. Dan hari ini kami memulai perjalanan tersebut dengan meluncurkan seri pertama, dasar dan fundamental, karena setiap data yang terpercaya lahir dari proses yang valid. Bersama membangun budaya mutu, meningkatkan kapasitas, dan menjaga kepercayaan terhadap setiap hasil pengujian dari laboratorium Badan POM untuk bangsa.
Thank you, ladies and gentlemen. Let us give another round of applause for this important milestone. This initiative reflects Indonesian FDA's commitment to enhancing laboratory competency. Your Excellency, the Chairperson of Indonesian FDA and the Permanent Secretary of Indonesian FDA, we may kindly invite you to remain on the stage. And we would also like to invite all deputies of the Indonesian FDA, the Principal Inspector of Indonesian FDA, Head of NQC-LDF, and Ibu Rita Endang to return to your respective seat. Thank you.
Ladies and gentlemen, we continue with the handover of guideline for quality assurance of laboratory testing result of the Indonesian FDA atau pedoman penjaminan kualitas hasil pengujian laboratorium Badan POM.
May we kindly invite the representative from Balai Besar POM di Jakarta, the representative from Balai Besar POM di Bandung, the representative from Balai Besar POM di Serang, the representative from Balai POM di Tangerang, and the representative from Balai POM di Bogor to come forward to receive the guideline, which will be presented by the chairperson of the Indonesian FDA. We kindly invite everyone to face the camera and smile for the official photograph. Okay, thank you. May this guideline further support the advancement of quality assurance practice within our laboratory. Your Excellency, the Chairperson of Indonesian FDA, the Permanent Secretary of Indonesian FDA, and the Head of NQC-LDF, we can kindly invite you to remain on the stage.
Ladies and gentlemen, we will now continue with the presentation of award to Balai Besar and Balai POM in recognition of their contribution to supporting the UMKM Paham Uji program. May we kindly invite the representative from Balai Besar POM di Samarinda, the representative from Balai POM di Bogor, the representative from Balai POM di Palopo, the representative from Balai POM di Payakumbuh, and the representative from Balai Besar POM di Jakarta to come forward to receive the certificate of recognition which will be presented by the chairperson of the Indonesian FDA. We kindly invite everyone to face the camera and smile for the official photograph. Thank you, Your Excellency, the Chairperson of the Indonesian FDA, the Permanent Secretary of Indonesian FDA, and the Head of the National Quality Control Laboratory of Drug and Food. May we kindly invite you to remain on the stage as we continue with the next part of today's agenda.
Distinguished guests, we will now witness the signing ceremony of the cooperation agreement between the Indonesian Food and Drug Authority and GP Pharmacy concerning support for the development of analytical method. This partnership reflects the importance of the ABG program, or academian, business, and government collaboration among regulatory authorities, industry, and scientific institutions in advancing laboratory capabilities. May I respectfully invite the representative from GP Pharmacy to the stage. Your Excellency Prof. Taruna Ikrar and the Permanent Secretary of Indonesian FDA, may we respectfully invite you to witness the signing ceremony of the cooperation agreement.
And may we now respectfully invite the representative of both parties to proceed with the signing of the cooperation agreement. Thank you. And now may we kindly invite the signatory to raise the signed agreement for the official photograph. Okay, thank you, ladies and gentlemen. Please join us in giving them a warm round of applause once again.
Thank you, Your Excellency, the Chairperson of Indonesian FDA, the Chairperson of the GP Pharmacy, the Permanent Secretary, and the Head of the NQC-LDF. May we kindly invite you to remain on the stage. Ladies and gentlemen, we shall now proceed to the group photo session. May I kindly invite honorable speakers, all deputies and the Principal Inspector of the Indonesian FDA, the Honorable Dr. Rita Endang, Apotheker MKes, Dr. Sofa Fajriah MSI, Profesor Dr. Ida Kusumawati MSI, and Professor Dr. Andes Bambang Kuswandi MSc PhD to come forward to the stage for the official group photograph in the first session. Thank you. We also invite the Honorable Fungsional Ahli Utama untuk dapat hadir ke atas panggung.
Okay, the first one is the formal pose. 3, 2, 1. Okay, the second one is checklist pose. 3, 2, 1. And the last one is semangat pose. 3, 2, 1. Thank you so much, Your Excellency, the Chairperson of the Indonesian FDA, and all distinguished guests. May we kindly invite you to step down from the stage and join all the participants at the front of the venue for the second session of the official photograph. Thank you.
Check.
And for all the participants, may we kindly you to join us for the official photograph.
Check.
You may come forward to
1,
the
2,
stage
3.
together.
Cek 1, 2. Ini kurang tinggi ya? Nah. Cek 1, 2.
Okay, the first pose is formal pose. 3, 2, 1.
GPF.
The second one is checklist pose. 3, 2, 1. And the last one is semangat pose. 3, 2, 1. Thank you so much.
Thank you very much.
Ladies and gentlemen, the official photo session has now concluded. Thank you, Your Excellency, the Chairperson of the Indonesian FDA, for your kind participation in group photo session. Distinguished guests, ladies and gentlemen, we will now proceed the next agenda, which the doorstop session by the Chairperson of the Indonesian FDA.
To that end, may we respectfully invite Prof. Taruna Ikrar, MD and Biomed PhD, accompanied by all speakers, the Permanent Secretary of the Indonesian FDA, Dr. Sofa Fajriah MSI, Mr. Tirto Kusnadi, Profesor Dr. Ida Kusumawati MSI, Profesor Dr. Andes Bambang Kuswandi MSc PhD, Head of the Center for National Quality Control Laboratory of Drug and Food, and Head of Bureau of Cooperation and Public Relations.
Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi rekan-rekan media sekalian. Very good morning to all of you. Terima kasih atas kehadirannya pada doorstep dalam rangka International Seminar Perspectives from 5 Continents, the Laboratory Future Sustainability in Innovation and Operations. Di depan rekan-rekan media telah hadir yang terhormat Kepala Badan Pengawas Obat dan Makanan Republik Indonesia, Bapak Profesor Dr. Taruna Ikrar, MD, M. Biomed PhD. Untuk itu dengan segala hormat kami mohon perkenan Bapak Kepala Badan POM untuk menyampaikan key messages terkait International Seminar on the Laboratory Future Sustainability in Innovation and Operations. Silakan Bapak.
Thank you everybody. Saya gunakan bahasa ya karena kelihatannya wartawannya Indonesia kan. Kawan-kawan, selamat pagi. Kami dari Badan Pengawas Obat dan Makanan melaksanakan sebuah kegiatan yang sangat penting. Kegiatan yang berhubungan dengan apa yang kita sebut dengan dari laboratorium yang bersifat innovation, sustainable, and interconnectednya dengan 5 benua. Nah, artinya bahwa kita tahu bahwa ilmu berkembang luar biasa. Yang kedua, alat-alat kesehatan khususnya alat-alat laboratorium juga mengalami kemajuan yang luar biasa. Jadi teknologi berkembang. Perpaduan antara teknologi dengan kemajuan tadi akan melahirkan banyak alat-alat baru. Taruhlah misalnya yang berhubungan dengan spektrofotometer. Dulu masih sifatnya spektrofotometer, sekarang sudah lebih tinggi dari itu. Dia sudah sampai penggunaan MRI, penggunaan kromatografi. Yang dulunya kromatografi tipe biasa, sekarang sudah sampai pada high performance, bahkan tiny scale yang sangat kecil. Dulunya kemampuannya bisa sampai mikro, kita sudah sampai kepada nano. Nah, kemajuan teknologi yang luar biasa seperti ini, tentu kita melihat karena dibarengi dengan tantangan yang luar biasa dari sekitar kita. Tantangan kita apa? Tantangan kesehatan. Kita melihat banyak obat-obat yang berkembang, tetapi banyak juga orang yang mau melakukan pelanggaran-pelanggaran, salah satunya obat-obat palsu. Demikian juga yang berhubungan dengan makanan, banyak melakukan tantangan-tantangan tersendiri untuk menghasilkan pengecekan, karena kan kami tugasnya memastikan keamanan, memastikan kualitas, dan memastikan apa yang disebut nilai gizi dan khasiat. Nah, dari kepastian-kepastian itu tentu harus didukung oleh apa yang kita sebut dengan tool atau alat, dan hari ini kita sudah mengembangkan perangkat yang lebih spesifik yang kita sebut dengan apps atau program khusus untuk penilaian obat dan makanan berbasis laboratorium. Kita sudah memiliki itu. Bahkan kami nanti lewat bantuan anggaran dari ADB, kita akan kembangkan lagi alat-alat laboratorium kita lebih luas lagi, dan bukan hanya di Jakarta tapi dari Sabang sampai Merauke. Nah, tetapi terkoneksinya Badan POM dengan laboratorium yang ada di Indonesia saja itu tidak cukup. Harus terkoneksi dengan dunia luar. Karena di situ akan muncul apa yang disebut dengan saling percaya. Nah, kami sudah tanda tangani misalnya confidential agreement dengan US FDA atau Amerika. Kami juga sudah punya kesepakatan dengan India. Kami punya kesepakatan dengan China. Kami punya kesepakatan yang sama dengan Jepang dan negara-negara sekitar, dengan Asian. Apa artinya? Kita pingin laboratorium kita juga terkonek secara global ke 5 benua. Kenapa? Karena produk dari 5 benua itu akan masuk ke Indonesia. Demikian juga produk kita akan disebar ke 5 benua. Jadi dengan interkoneksi seperti ini, endingnya adalah pertama kita mengembangkan sistem inovasi yang kita sebut dengan Green Laboratorium. Yang kedua, tentu dampaknya yang paling keras, yaitu apa yang kita sebut kapasitas dan reputasi laboratorium kita semakin diakui. Dan ending terakhirnya kita berharap masyarakat dengan reputasi yang dimiliki laboratorium-laboratorium kita, maka kepercayaan masyarakat terhadap produk yang beredar di tengah-tengah mereka semakin tinggi, sehingga dia merasa lebih aman, merasa terjamin keamanannya, terjamin kesehatannya, terjamin khasiat dan kualitas yang dia konsumsi. Jadi inti akhir daripada kegiatan ini yaitu bagaimana Badan POM menunjukkan tekadnya untuk mengayomi dan sekaligus melindungi masyarakat seluruh Indonesia. Saya kira itu inti utama acara hari ini. Silakan kalau ada yang nanya.
Ya, terima kasih banyak Bapak Kepala Badan POM. Now we have the honor to kindly invite Dr. Tien Do, Secretary General of the High Performance Thin Layer Chromatography Association International, to give a few words on the international seminar and the collaboration between HPTLC and Indonesia to our media colleagues.
Hi, good morning. Thank you for the invitation. For me, it's an honor to be here and be part of your seminar. The HPTLC International Association is about collaboration. So we are clearly part of this seminar today. And the goal of the association is really to collaborate within the world, the entire planet, working on HPTLC and see how HPTLC can be used for the safety, the quality, and the efficacy of all the products that are sold around the world. So for Indonesia, it's all the products that are sold inside your country are also important to your country, and we would like to collaborate with you to see how we can help you to go into that direction for the safety of the consumer and for your country. So as part of many different expert committees from the USP, the European Pharmacopoeia, or the African Herbal Pharmacopoeia, we have an overview of what is going on around the world for all the products. And we clearly see that there is more and more adulteration product on the market. And that's why the goal of working all together and collaborate on developing methods, innovation, on technology, it's really useful. So we are happy to be here and share with you this day on the technology, innovation, and safety of all the product. Thank you.
Thank you very much, Dr. Tien Do. Now we would like to open the floor to our media colleagues for questions. Silahkan rekan-rekan media untuk, silahkan Mas dari detik.com.
Ya, selamat pagi Prof. Taruna, saya Vers dari detik.com. Mungkin ada dua hal yang ingin saya tanyakan. Pertama terkait mungkin arus produk obat, suplemen, dan pangan itu kan yang dari luar negeri sekarang semakin banyak ya Pak, semakin makin berjamuran gitu. Dari BPOM sendiri sebenarnya bagaimana kapasitas dari lab di Indonesia sendiri, situasinya seperti apa? Mungkin apakah ada kendala, apakah sudah memadai? Dan mungkin bagaimana caranya dari BPOM sendiri untuk membuat itu bisa maksimal. Terus yang kedua tadi dalam paparan Prof. Taruna menyinggung masalah soal mikroplastik yang pada saat ini parameternya masih belum jelas bahkan di seluruh dunia. Mungkin bisa dijelaskan Pak, kenapa parameter ini diperlukan, mungkin apa concern bagi BPOM dan mungkin resiko kesehatan seperti apa. Terima kasih.
Terima kasih detik.com. Yang lainnya media dari El Shinta, RRI, sudah cukup? Silahkan Bapak Kepala Badan POM.
Izin Pak. Ya. Ada dua pertanyaannya. Yang pertama tentu kita sesuai dengan kewenangan kami, Badan Pengawas Obat dan Makanan, yang memang spektrumnya sangat luas. Kewenangan Badan POM itu mulai dari riset development sampai law enforcement, pre-market sampai post-market. Kita tahu sekarang memang sangat besar produk-produk yang kita impor. Kalau kita berbicara tentang obat, masih 90% bahan baku obat kita impor. Nah, bahan baku itu terbagi dua, ada bahan baku yang betul-betul raw material, ada juga yang intermediate product. Yang kedua, bicara kosmetik, masih mayoritas, kalau kosmetik kita mayoritas 80% itu industri dalam negeri. Memang hampir 20% ada produk-produk multinasional. Nah, jadi suplemen juga seperti itu. Kita suplemen sudah memiliki banyak produsen dalam negeri, tetapi karena dia biasanya terkait dengan produk-produk obat, itu biasanya masih multi-center atau multi-country, multinasional. Nah, kemudian makanan. Makanan kita tidak bisa pungkiri bahwa makanan itu terbagi dua. Ada yang merupakan produk-produk dalam negeri yang memang mayoritas masih kita produksi di dalam negeri. Tapi ada beberapa makanan-makanan yang kita impor. Nah, sekarang hubungannya dengan kesiapan Badan POM sebagai laboratorium pengetes. tentu sampai sekarang ini kita termasuk yang masih terbaik di Asia Tenggara khususnya, karena kita punya laboratorium dalam hal pengujian, apakah itu pengujian makanan, pengujian obat, pengujian narkotik, pengujian tobacco atau sigaret ya, kemudian pengujian, bahkan pengujian tertentu misalnya obat-obat yang berhubungan dengan virus dan sebagainya. Kita punya animal center yang levelnya cukup tinggi. Nah, dengan konteks itu fasilitas laboratorium kita siap menghadapi secanggih apapun produk, termasuk produk-produk obat yang bersifat inovatif, produk-produk baru. Jadi kita tegaskan bahwa Badan POM dengan kelengkapan laboratorium dengan sistem regionalisasi yang kita miliki, dengan alat-alat canggih yang kita miliki, kita siap beradaptasi dan siap menjalankan tugas itu, termasuk memproteksi rakyat kita dari berbagai macam produk-produk yang kemungkinan punya efek bahaya kepada masyarakat. Jadi kalau ditanya kesiapan laboratorium kami siap untuk itu. Nah, sekarang yang terakhir, yang berhubungan dengan mikroplastik dan nanoplastik. Kita tahu seperti yang saya mention bahwa sudah bukan hanya di New England Journal, ada juga publish di Science, ada juga publish yang di American Circulation Journal, yang sifatnya sangat spesifik bahwa mikroplastik sudah menjadi ancaman. Kita dengar beberapa saat yang lalu hujan kita saja, our rain itu mengandung kandungan mikroplastik, dan itu kan sempat heboh di masyarakat. Nah, sekarang tentu itu kenyataan, dan salah satu penyebabnya mikroplastik ini asal muasalnya dari sampah-sampah yang mungkin karena proses degradasi dia terbang ke atas menjadi debu. Ya, kemudian dia bisa meresap ke bawah ke air dan ikut sama air dan kita minum, kan begitu. Jadi kesimpulannya, apa bedanya mikroplastik? Kalau mikroplastik itu sebetulnya adalah ukuran daripada, kan turunan plastik banyak ya. Nah, dari turunan itu yang jelas molekulnya yang lebih kecil dari 1 nanometer, itulah yang disebut dengan nanopartikel. Kalau ukurannya lebih kecil dari 1 mikrometer, itu namanya mikropartikel. Nah, dari kedua hal itu, ya ini fakta cukup mengkhawatirkan. Nah, walaupun tentu case-nya masih kita perlu evaluasi, kita tidak mau membuat rakyat kita panik. Nah, tapi masalahnya secara global belum ada standar pun, baik yaitu kodeks kita punya organisasi dari FAO ya, kemudian kita juga dari WHO, kemudian standar global belum ada bahwa berapa kandungan mikroplastik atau nanoplastik yang diizinkan, itu belum ada. Nah, oleh karena itu lewat kemarin di Komisi 7, waktu kami hearing dengan parlemen, kami dikasih tugas atas nama rakyat, tentunya mereka meminta bahwa Badan POM akan bekerja untuk memberikan skala tersebut. Mudah-mudahan dari skala yang dikeluarkan oleh Badan POM nanti bisa menjadi masukan secara global. Jadi, dan itu kan perlu disepakati nanti. Jadi sekarang kita on progress untuk memberikan scale yang berapa yang cukup aman. Kan kita kalau untuk yang sifatnya bisphenol A, kita sudah punya ukuran-ukuran, bahkan saya sudah buatkan keputusan. Tapi khusus untuk sifatnya plastik, ini belum ada ukuran yang pasti. Nah, sekarang tim kami dari P3OM dengan laboratorium yang kami miliki akan melakukan, membuat dan melakukan skala. Tentu berdasarkan berbagai referensi, khususnya referensi dari riset-riset yang terstandar ya. Seperti yang publish di Nature, New England Journal, pasti itu menjadi landasan referensi kami. Terima kasih.
Terima kasih Bapak Kepala Badan POM. Cukup ya rekan-rekan media, Bapak sudah ada agenda lain. Terima kasih.
Harus berikan.
Ya. Terima kasih banyak Bapak Kepala Badan POM. Thank you very much Dr. Tiendo, as well as our distinguished guest speakers, as well as BRIN and BPOM representatives. Demikian rekan-rekan media, doorstep dalam rangka International Seminar Perspectives from Five Continents, the Laboratory Future, Sustainability in Innovation and Operations. Terima kasih. Wassalamualaikum warahmatullahi wabarakatuh.
Ladies and gentlemen, thank you very much for still in this room. Actually, we will begin the first session of our international seminar at 9:30. And now we would like you to enjoy your coffee break first. Thank you.
If I do that, but if I do this, can you tell me if you see this or you see the sharing? Okay, I will see the monitor.
Okay, distinguished guests, ladies and gentlemen, as we conclude the ceremonial session, we now enter the scientific component of today's seminar, where knowledge, innovation, and collaboration come together to shape the laboratories of the future. Through the scientific session that follow, we will explore how innovation, digital transformation, sustainability, and advanced analytical approaches can strengthen laboratory performance and support evidence-based regulatory decision-making. More importantly, this session provides an opportunity to exchange experiences, foster international collaboration, and inspire new ideas that will contribute to the advancement of laboratory science and public health. With the shared commitment to scientific excellency and sustainable innovation, we shall now commence the scientific session of today's international seminar.
Ladies and gentlemen, we shall now proceed to session 1, entitled High Performance Thin Layer Chromatography or HPTLC. As analytical sciences continue to evolve, innovative technologies play an increasingly important role in enhancing laboratory efficiency, reliability, and analytical capabilities. Among these advancements, HPTLC has emerged as a versatile and powerful analytical technique with broad application in pharmaceutical, herbal, and food analysis. This session will provide valuable insights into recent developments and applications of HPTLC in modern laboratory practices, contributing to improved analytical performance and scientific excellence. This session will be presented by Dr. Tiendo, the Secretary General of the HPTLC Association International.
Without further ado, I would like also to invite our moderator, Professor Dr. Apoteker Ida Kusumawati MSI, to lead the session. To our speakers and moderator, the floor is yours.
Okay, Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. Thank you for nice introduction for the Master of Ceremony. Very warm welcome to all distinguished speakers and participants today. It is my pleasure to serve as the moderator in this session. It is now my great honor to introduce our distinguished official, my best friend, Dr. Tindu. Dr. Tindu is the Secretary General of the International SPTLC Association and also the Chief Scientific Officer at CAMAC, Switzerland. She holds the PhD in chemistry and has extensive expertise in analytical chemistry, particularly in the development and application of SPTLC method for natural product and pharmaceutical analysis. In addition to her leadership and research contribution, Dr. Do also as an expert volunteer of the United States Pharmacopoeia and is a member of expert group for the both European Pharmacopoeia and the Swiss Pharmacopoeia. She also contributes as an expert to ISO Technical Committee 249, focusing on traditional Chinese medicine. Before I invite the doctor to begin her presentation, I would like to share one brief reflection. I believe many of us are familiar with chromatographic techniques such as SPLC or GC. But when it comes to HPTLC, many people still underestimate the capability or may not yet realize how much this technology has evolved over the years. Today, SPTLC is no longer just a complementary analytical technique or just for screening methods. It has become an increasingly powerful tool for quality control for herbal product. That is exactly why I believe today's presentation will be especially valuable for all of us. And now with that, it's my great pleasure to invite Dr. Ju to share her expertise on SPTLC, the new front line of global botanical surveillance. Dr. Du, time is yours.
Thank you for the nice introduction. So it's good morning, everyone, and it's an honor for me to be here today and talk about HPTLC. But the first thing I would like you to think, so if we can share the presentation now. Yeah. So I would like you to imagine that you are a regulator or a pharmacist or a quality control analyst. You have a botanical product that will arrive in your lab. And immediately, 3 simple questions appear. First, what plant is in your product? Second, is it really the right species or just a look-like? And third, maybe the most importantly, is it safe? Now, in theory, these questions are straightforward, but in reality, they are not. Because now, imagine that you don't have only one sample, but hundreds. Limited staff, limited time, limited analytical capability. So the real question becomes, how do you make reliable decisions fast enough? The botanical supplement market is already worth tens of billions of dollars. Thousands of products, hundreds of plant species, and a supply chain that spans the entire planet. But regulators and laboratories still face a very simple question: what is really inside the product? Misidentification, adulteration, and safety issues are not theoretical risks. They are daily analytical challenges. In this context, laboratories need fast, reliable, and scalable tools. So my name is Tin, and today I will show you how modern HPTLC is emerging as a new front line of botanical surveillance. Now imagine this is you, and it is your lab. You are expected to answer multiple questions at the same time: identity, purity, and safety. But at the same time, the number of samples is increasing, and resources are limited. This is not a rare problem. It is happening everywhere. Botanical quality control is approaching a breaking point. On top of that, we see increasing cases of species adulteration, species substitution, and variability in composition. And importantly, these issues are not always detectable with targeted methods. So the challenge is clear. How do we screen efficiently without missing critical information? So how do we deal with this complexity? Traditional analysis often focus, like here, on one compound. So here you see routine. But complex sample, we have hidden risk and limited visibility. And this, it's what we see in HPTLC. We see a holistic chemical fingerprint. Each track represents a sample, and each band reflects a part of its composition. We don't look at one compound anymore. We look at the entire chemical profile. But what does it mean in practice? What can we do with this information? Let's take a real sample now. We start with a simple question. Is it a right species? Here, we will compare reference material here, and we have commercial sample on the right side. At the first glance, some may look familiar, like here. They all look similar, they are compliant. But some, like here, do not match the fingerprint. we can see some additional zone or some missing zone. So with one HPTLC plate, we can immediately detect species substitution. And it's the case here. Instead of having a ginkgo biloba leaf, we have a sophora fruit. So here we show that HPTLC can assess identity. But the same plate can tell us much more. HPTLC does not just generate data, it generates evidence you can see. Now let's ask another question. Is this product pure? Using the same case study here. The reference here shows that a characteristic pattern. And here we clearly see that in this sample we have additional zone. Here in this case, ginkgo biloba is adulterated with sophora. And you clearly see the additional zone here. So we indicate the presence of another ingredient. So with one HPTLC plate, here we can detect adulteration. Same as here, we can see that the extract it's not a raw material, but it is an extract because the zone is more intense. So HPTLC does not just generate data, it detects adulteration. And beyond purity, what can we assess? So let's consider safety now. Is this product safe? Here, we analyze the same ginkgo biloba product sample using another specific method. You know that certain compounds must be strictly controlled due to their toxicity. Here, it's what we observe with the ginkgolic acid. These compounds are known to be allergenic and must be limited. So with this method, we can really clearly see the intensity and see if they are present or not. Other possibility are the bioassay, directly on the plate. Here, it's an example of the planar yeast heterogeneity screening, so the PES test. Here, with this test, we can see toxicity of compounds such as estrogen, estradiol, and other compounds. So you see that here, HPTLC, we can directly detect the safety risk. We can detect when there is allergenic compound, and we can also detect when there is toxic compounds directly on the plate. You have seen 3 examples that answer 3 simple questions, but together they do something more. What we have seen so far is complex chemical information. But in practice, analysts don't need complexity. They need decision. Is it the right species? Is it pure? And is it safe? This is what transforms HPTLC from an analytical technique into a decision-making tools. You can see on the screen here, you have the reference on the left side, and then you have sample on the right side. And you see that the software can flag all the parts in the fingerprint that are different from the reference material. So you will see immediately if one sample is not compliant. So here we have identity, purity, and safety. In HPTLC, we don't need to identify everything. We just need to know when something is wrong. Another case here is the detection of synthetic drugs in dietary supplement. We know that more and more products on the market are supposed to be natural, but actually they are not. And many suppliers are adding some drugs inside dietary supplement. So it's the case of a lot of weight loss products or live product balance that are seen on the market, that you can buy on internet in different parts of the world. So here, it's a project where we work with the FDA in US and they send us many different products that they can buy on the internet. And what we have done here, for example, with the case of the PDE5 inhibitors, for example, looking at sildenafil citrate, but not only sildenafil citrate. So we analyze all the samples that we have received, and then we use the same tool. So we look at the pattern here. We are supposed to see nothing in the product because it's natural, so you should detect nothing. But then when you look into the HPTLC chromatogram here, you can clearly see that some of the products here are not compliant, and they contain zones that should not be here. So here, in that case, we look at sildenafil citrate, acetyl sildenafil, and other analogs. So here, with this tool, it's the same. We have a decision tool making. We are looking at identity, purity, and safety of the product. If we look at other drugs and forensic drugs, for example, we have here one example where you can see different compounds like ketamine, methamphetamine, cocaine, heroin, amphetamine. You can see that we have different drugs on the plate, and we can clearly detect them. They have different RF value, different position, they have different colors. And then even if you do more testing on that, you see that we can have RF value, UV spectra, and we can combine all that to MS and analyze the MS directly on the plate. Another case where it's emerging, it's, for example, etomidate in illicit market. Since few years, we have seen a growing non-medical use of etomidate in Asia and New Zealand, especially in vaping, electronic cigarette. So we have many different cases in Thailand or in Singapore where now electronic cigarettes are banned because we have seen that many people are using e-liquid and put some drugs inside. So because of that, we use HPTLC, and you see here some application note, where we detect nicotine, but also etomidate inside. The method is fast. Within less than 30 minutes, you can analyze more than 15 samples in the same time and see if they contain etomidate. Here is what you see. We have a spectrum here, and we can clearly separate etomidate from nicotine. You have to know that etomidate is not the only drugs that is used in electronic cigarette. So the method will be expanded to different drugs. For example, the drugs that you have seen before. So this is the goal of HPTLC, to expand each method now to non-targeted screening for the drugs. Another case is the anti-malarial drugs. For example, in the European Pharmacopoeia, we have several methods for all those different drugs. So you see that here we simplify everything, and we have only one method for the identification of the 14 anti-malarial drugs. They are clearly separated, and you can do the UV spectrum to identify them. So HPTLC is really easy to identify drugs now. What you have to remember is HPTLC is not TLC. So you have to forget about TLC. What we are doing is HPTLC. HPTLC, we had the evolution of the instrument. So we had, before we were working manually, nothing was really standardized. Now we have some instrument on the market that can be automated or fully automated. But the main evolution is the software. The software brings some quantitative information in HPTLC. What you had before for quantification, we were applying different standards at different application volume to have intensity. And what we were doing before, when we had sample, it was just to estimate. We did a visual estimation of the quantity. So we were just moving the sample around the calibration curve to know where it was. So this was just estimation. Now we had an evolution in the software. What we have is, with the software, we have one fingerprint here. The software is looking 5% below the application position and 5% above the solvent front. Then we'll look at the center of each track. From that, for each pixel, it's calculating the RGB value. From the RGB value, the red, green, and blue value, it's calculating the luminance with this equation here. And when you are calculating the luminance from that, then you can plot the luminance against the RF value. And in that case, HPTLC is becoming quantitative. You have intensity value, so you have quantity. What we were using before for quantification was the scanning densitometry. So the scanning densitometry is using different lamps, different mirror, and then it's measuring on the plate. Here, it's one result of the scanning densitometry, where you can scan at different wavelengths, specific to, for example, here, for each color. So if you think, again, about quantification that we were doing the visual estimation before, now, with HPTLC, it's different. You have your plate, you have the calibration standard here on the plate, and you have your sample here. From that, you can generate peak profile from images or peak profile from scanning densitometry. And from that, you will generate the calibration curve. When you have a calibration curve, then you can do the quantification of your compound inside. So you see, quantification can be done from the images or from the scanning densitometry. From the images, it will be less precise than the scanning densitometry, because images is based just on 3 channels, and it's depending on the quality of your picture. With the scanning densitometry, you are specific to a wavelength, so you are more precise. But still, you can still do quantification in both cases. So now that you know that HPTLC is also quantitative, for one sample, you see that we have multiple information. We have HPTLC images. You can have picture of your plate before and after derivatization. You can also combine different derivatization. Then from each images, you will transfer into peak profile from images. Then you will have intensity information. And then if you add on top of that the scanning densitometry, then you have much more information. So you see one sample, and here in that case, we have 9 different detections. Everything is just in one analysis. What you have to know now is that HPTLC is already used. It's not a future concept. It is already integrated into major pharmacopoeias. We have the United States pharmacopoeia, we have the Indian pharmacopoeia, or you also have European pharmacopoeia. The latest one is the international pharmacopoeia supported by the WHO. Since this year, we have a general chapter on HPTLC. We have also, what is new since 2026, is the publication of the African herbal pharmacopoeia using HPTLC for the identification part. So this shows how HPTLC is already supporting decision-making worldwide. So the question now is no longer whether HPTLC works, it is whether you choose to use it. In regulation, HPTLC means standardization. Standardization of each step. Standardization to be reproducible, to be able to compare all your data from plate to plate, lab to lab, year to year. The first part of the standardization is the plate and the plate layout. The regulation mentions how you have to apply on the plate, where to apply on the plate. And you have to follow that if you want to be compliant to those pharmacopoeia. The other thing is also how to apply on the plate. To apply a spot or a band, using a contact or a spray-on. The regulation will recommend here to apply by band and by spray-on. Why? It's because when you are spraying on the plate and you are not touching the layer, and when you are touching the layer, you are first interfering with the layer, and you have also diffusion. And you see on the left side here, you have diffusion on the plate already. And when you have diffusion at the application position, you will have diffusion during the chromatography. So the best is always to spray your sample on the plate and not touch the layer. Like this, you only have a precise spot, and then you will have a chromatogram without diffusion. The length of the lens, the length of the band, sorry. So here, the regulation will recommend to apply a 8 millimeter band. Why? It's because when it's too small, it's complicated to describe your sequence of zone. When it's too big, you are losing space on the plate. So the regulation will always recommend to do 8 millimeter. For the developing distance, it's the same. The regulation here will recommend to have 7 centimeter for the developing distance. Why? It's because when it's too small, you don't have a good resolution. And when it's too long, it will take more time. So sometimes it's not useful to develop more. So they will recommend to have 7 centimeter. What is important if you want to have reproducible result is to standardize and do all the time the same thing with the chamber configuration. In the environment, we have humidity. Here, the humidity is higher than what we have in Europe. For example, you can have sometimes 80% humidity, and in our case, we will have 50% humidity. The humidity will affect the RF value, the position of your band. So you have to control the humidity on the plate. You also have to control is the saturation, the equilibrium that is inside your chamber. Are you saturating? Are you doing unsaturated? Or are you preconditioned the chamber? In the regulation, they will mention to do saturated. Why? It's because if you have a higher temperature, the evaporation of the mobile phase, you will have evaporation of solvent in your chamber. And with higher temperature, the evaporation is faster. Your temperature, if the lab is not controlled, will increase this evaporation. So to be sure that we all have, no matter the difference of temperature, that we all have the same equilibrium in the chamber, the regulation recommends to all saturate our chamber. And like this, we are all at the same step. That's why here, you see, on the plate here, we have different results if we don't saturate the chamber and if we saturate. If we all do the saturation for 20 minutes, we will all succeed to have the same RF value. That's why it's important to control the saturation. The last part here is the humidity. I was telling you, in Europe, we don't have the same humidity as here in Indonesia, for example. And you see here we have different examples where, in the first example, on the top left, the humidity will affect the RF value, no matter which humidity. In some case, the humidity is not affected the RF value at some point, but you have to show where is the limit of your method. The humidity can also affect the diffusion on the plate, not only the RF value. So it is really important to control it. You will see controlling all the parameters can be done manually or with instrument. So you will see during the workshop, there is different case where you will do the standardization manually without instrument. And you will see it's possible. That's why HPTLC is part of all the regulation in the pharmacopoeia, because it can be done manually, it can be done with instrument. It's accessible, and for the user, it's really easy to do. HPTLC is also a technique that is qualified. For that, we use SST, System Suitability Test. So in HPTLC, each plate is qualified. We need to perform SST on each plate, each analysis. The SST is here to verify the analytical method. You have to prove that you did everything in a good way. The SST is for each specific method, each mobile phase, and you will have defined acceptance criteria. If the SST is not passing, the analysis fails, and you cannot use the data for the evaluation. What is...
Sorry to interrupt.
Sorry.
So we have the national anthem of Indonesia. Please rise. Please be seated. Thank you.
So we were saying that in HPTLC we have critical parameters. So the first thing is the plate. You have different supplier of plate. There is Merck, Nagel, there is other supplier. For example, we have Chinese plate, silica plate from Canada, or we have many different suppliers. You have also different type of plate. We have reverse phase, amino plate, diol plate, cyano plate. What is important when you start to develop method in HPTLC is when you start with one supplier, you have to stay with the same supplier, because they don't use the same recipe for the plate. They will have different selectivity. So you have to know if, when you test your method, you have to know what is the limitation, which supplier you can use, which one you cannot use when you develop this method. It is really important. Then you have the development. I told you the first thing is the humidity, the activation step. Then we say the saturation. You have to control your saturation. And then the developing solvent. How are you doing your mobile phase? Are you sure that you did the proportion in a good way? It happens that sometimes we do some mistake. So you have to be sure about what you have done. Currently, in pharmacopoeia, they are using SST, system suitability test. And for that, you have two examples here, using the same plant species, St. John's wort. On the left side, you have the USP method. And the USP method for the SST will look at the reference extract and will look at one part in the bottom of the chromatogram and one other part with the red zone in the middle of the chromatogram. On the right side, you have the European pharmacopoeia. And the European pharmacopoeia for the SST is just looking at the lower part of the chromatogram. So you see, both USP and European pharmacopoeia are just looking at a specific region of the chromatogram. But us, we know that when there is a problem in the gas phase, in HPTLC, it will touch the upper part of the chromatogram. So here, we see that the problem of both pharmacopoeia is that they are not looking at the entire chromatogram. So they are missing information. The problem also with the current SST is that for each method, we need to do specific solution for the SST. So it means that we have to all the time prepare fresh standard with reference standard. And at the end, doing fresh standard all the time, it's quite expensive. Not only expensive, it's taking a lot of time because we have to all the time prepare fresh solution and we have to make sure that we have the standard in our lab. So at one point, we had some requests from user. And the idea of having a general SST came from India. They wanted to have a more practicable solution for HPTLC, for routine analysis. So for that, they asked us to have a more holistic approach, to have a kind of universal mixture of substance that you will use all the time for all HPTLC method. And this solution will qualify the entire chromatogram. How we did that is that we start a project with Sigma. They give us a list of more than 1,000 compounds from their catalog. From this 1,000 compound list, we set some criteria. What was important for us is that all the compounds that we will select has to be detectable without derivatization. It means that you need to be able to see them in white light, for example, in 254, or in 366 without derivatization. What is important also is that all the compounds selected has to be of minimal hazard. We want the user to be safe and not use toxic compound. The solution has to be also stable for at least 2 months in solution. Because the goal is not to prepare fresh all the time. We don't want to lose time. Time is money, so we have to be a bit more fast. Then it has to be available. It means that you need to be able to have here the compounds, but also in US, in Europe, and in Africa. We don't want part of the world that have no access to the compounds. And most importantly, it has to be not expensive. We don't want to spend too much money on a system stability test. So at the end, from the 1,000 of compounds from the list, we reach 8 compounds. The 8 compounds are here. So you see, you have the name, but you see that they have different groups. They are from different polarity and different selectivity. So we have now one solution of 8 components inside that we call the UHM, the Universal HPTLC Mix. We have tested this UHM in more than 20 different mobile phases of different polarity and different selectivity. Here are some examples. And you see that no matter which mobile phase we are doing, we all the time have a fixed sequence of zone. We have at least 3 to 4 zones in the entire chromatogram. And it's how we are qualifying our plate. What does it mean? It means that if, for example, you are repeating the mobile phase 2 here, you need to all the time having the zone D at the same position. You need to have the zone E at the same position. Same for F, G, and H. They have to be at the same position all the time. If they are not, it means that you did something wrong. You didn't control the activation, the humidity on your plate, or you didn't control the saturation of your chamber, or you did something wrong with your mobile phase. You need to all the time have this sequence of zone for each mobile phase. So I told you, the UHM is here to check that everything went well. So it means that we have to be able to detect when something is wrong. The first case is the humidity. If now you forget to control the humidity on your plate. You see that with this example, the mobile phase 5, we have different sequence of zone depending on the humidity. If you are doing 0% humidity, 35, 75, 90% humidity. So if you forget to put the activation of your plate, or you forget to put your plate in the desiccator to control the humidity, you will have different RF. You will have different position. And here you will immediately know that you cannot use the plate for the evaluation later. Here, same thing. If you are doing here the mobile phase in the wrong way, here it's the mobile phase number 6, and you see that here we did different tests, plus minus 10% of the ratio. We have different RF value, but we also have different delta between the zone. So we can easily detect when something is wrong with the mobile phase. Last thing, saturation. If you forget to saturate your chamber, then you will have completely different result. And even when you forget to put the filter paper in the chamber, you will have different results. So we are able to detect wrong humidity, wrong mobile phase, or improper saturation. We did this test in different countries. We send this solution, the UHM, we send it to US, India, and France. And they did the same test. And we were able to have all the same RF value with a deviation that was no more than 0.04. In practice, the software will check the SST all the time before the evaluation. So you will have value that you obtain during the method development and during the validation of your method. You will have the error value, and you will apply the delta, the deviation that you have calculated. And then it will look at the position, and if he can detect the zone at each position, then the plate will be compliant. And when the plate is compliant, then you can start the evaluation. So you see, so far, HPTLC can do multiple things. But how we can use that in practice? Here we have automated plate development, followed by imaging under different conditions. HPTLC combines multiple detection modes: UV, visible light, derivatization. And all the time we have intensity. So you see, with one plate, one sample, we have many information. So HPTLC is no longer a manual technique. It is standardized, reproducible, and has a really standardized workflow. But with the advanced software tools, we have much more. we can now move from images to data, and to data to quantification. One of the key challenges in botanical fingerprint is we cannot do a simple yes or no comparison. It's not sufficient. Here you see different samples of ginkgo. They are all good, but you see the fingerprint are also different. This is natural variability. It's something that we know in botanical. Samples can come from different locations, depending how they were harvested, how they were stored, you will see different intensity. So how we do that in HPTLC to address natural variability? With advanced tool, we can now define reference criteria, acceptance range. So we will have a pool reference here. We have a pool reference profile here. And from that, we have acceptance criteria. So what you see in pink is the acceptance, which sample we will accept. If one of the sample has intensity that is higher than this pink area or lower than this pink area, we will not accept that. So the variability now of the sample is not a problem, but the lack of structure interpretation is. So far, in HPTLC, we interpret HPTLC fingerprint visually, and we look at individual band, and we compare sample. But this is interpretation. Now, here you have 10 different batches of polyherbal formulation. In different countries in Asia, we have many products that contain not just one ingredient, multiple. So how we interpret multiple formulation now? We use chemometrics approaches. So with chemometrics approaches, we no longer look at individual band. We look at the entire fingerprint as a data set. We are not just looking at differences, but we understand patterns. This allows us to classify sample based on similarity. Here, it's what we are seeing here. For this sample, this product contains 4 different ingredients. You have here the fingerprint of the 4 ingredients. We create a model in machine learning using these 4 ingredients, so we have specific pattern for each of them. And then we have here our 10 products. And when we use the machine learning and the model that we have developed, we are able to qualify the finished product now. And you see on the screen, we have the score for each ingredient. And when you look at the 10 batches that we have, they have different value, but it's all the time the same range. For example, with the ingredient 1 in the finished product, the score is always between 11 and 14 or 17%. Even if the numbers are not all the time the same, it is normal. It's the natural variability. But they are from the same range. If, for example, one of the batches would have 3%, we would see that this batches is not compliant. So with chemometrics now, it's really easy to characterize and qualify polyherbal formulation. So we have now a powerful tool for that. Another example, Asian ginseng and American ginseng. Here, you have an example of 38 samples. We have different detection modes. So here we are using 2 different detection modes after derivatization. And the goal here was to create a model. We had a model based on those samples. So we create a model with 15 samples of Asian ginseng and 15 samples of American ginseng. We see we have different natural variability. We have different points on our PCA here. And then we had 8 new samples. 8 new samples, and we don't know what they are, if they are Asian ginseng or American ginseng. Using our model, we know where they are now. We know where they are positioned, and you see that here we have a list of our 8 new samples, and 4 of them are Asian ginseng, and 3 of them here are American ginseng. And you can see that in the graph here, or we have also a table with scoring. So you have value. And for example, we have here the 5 samples here on the top are Asian ginseng, and the score is between 91 to 96.5% of confidence. The same thing for the 3 last samples that were qualified as American ginseng. So now we are not just looking at specific marker of ginsenoside, because it's too difficult. People can add the specific ginsenoside just to say that it's Asian ginseng. So if you look not at specific marker, but look at the entire chromatogram, then you are able to qualify everything now. So this is how we are working now. We are looking at the entire profile, and the software is telling us which species it is and how confident it is. There is a value. So you see, we are not just seeing differences. We understand pattern now in HPTLC. But this will lead us to the next step. Each sample will generate a fingerprint that can be stored, compared, reused. Over the time, we are building a big database. We compare based on similarity, and we use to predict similarity and identity. So the fingerprint is becoming a digital identity. Using artificial intelligence coupled to HPTLC, we can now have prediction tool. So what comes next? As we build larger database, the more data we collect, the more powerful the system becomes. This opened the door to new capabilities. We are now able to predict error value. We are now able to have similarity matching. And now the next step will be the species prediction. The goal of having HPTLC with digital transformation is to be able to have a fingerprint, and the software will tell you which species it is. And if there is mixture, it will tell you which mixture it can be. For that, we have now new concept. We work into the non-targeted screening. You have seen that around the world, that many different countries have different authorities, agencies, to control the quality of herbal product. You have here different examples of different countries. So all of those authorities will control the quality of your product. But at the end, what they want is when you buy an apple, what you want to be sure is that you have a good apple. You want to be sure that you are not buying an apple of poor quality. And the worst case is that you don't want to buy an apple that looks like an apple, but actually it's not. You need to have proper identification tools and specification for the quality of your product. That's why HPTLC is mostly used for identification. Here, one other example. We have Vitania somnifera, ashwagandha roots. So you know about this root. We have in the literature or in the regulation different analytical methods here using HPTLC or TLC. There is many methods available. So when you start, which one you will select? Which one is the best? There is so many methods outside that you don't know how to start now. So that's why here we work now with a new concept that is called the Complementary Developing Solvent, the CDS. How it works is that we have now a non-targeted screening approach. Each sample will be tested with 3 different mobile phases. So we have here the low polar developing solvent, the medium polar developing solvent, and the high polar developing solvent. Each developing solvent has different polarity and different selectivity. And we were thinking that by combining all the results, we will have a specific fingerprint for each sample. Here, you have example of the green tea, the olive leaf, or the peppermint. We have the result with the 3 different mobile phases, and we combine the fingerprint together here. And then, when you compare to the specific method of the European Pharmacopoeia or the United States Pharmacopoeia, you see that the 3 mobile phase concept give us all the time more information. And you can be sure that not 2 different species will have the same fingerprint. So what we thought in theory is actually working. With the 3 mobile phase concept, we are able to have a specific fingerprint for each sample. In practice, if we're going back to the ashwagandha example, you had the USP method here, and the USP method give you that for the result. You had the roots, the leaf, the small roots, and we have some standard here. To develop this method for the USP, it took quite a long time. But if now we test with the 3 mobile phase concept, you see that we had exactly the same result. We can discriminate roots, leaf, and small roots also without optimization. We use only 3 mobile phase. We don't optimize. It's simple. So you have the result here with the low, the medium, and the high. And you compare to the USP. We have much more information with the complementary developing solvent. The complementary developing solvent concept is also qualified using the UHM. When you look at the benefit of the non-targeted screening, when we look at adulteration, I told you adulteration is now more and more present on the market. Here is the example of the ginkgo with the sophora. We have on the left side the USP method, and you see that the USP method, we can detect zone of adulteration. So you have the ginkgo on the left, sophora on the right, and in the middle you have a mixture of 20% adulteration. When we developed this method for the USP, it took at least 6 months to have a good method with specific marker for adulteration. But when you look at the 3 mobile phase concept, without optimization, we are able to detect specific marker too. So you see that now we don't lose time in developing method. When we used the 3 mobile phase concept, we have immediately our answer. We can detect positive marker of adulteration in our product. You can also look at specific compounds, phytochemical classes. Here we did the test with 3 different classes, coumarin, iridoid, carbohydrate, and you can plot them in 3D with their RF value. And you see that they can separate easily. So it means that when you have a new sample, a new compound that you don't know, you will do the 3 mobile phase concept, and then you can plot this in 3 dimensions here. And you will see where it is, which compound it could be, which phytochemical classes it could be. When we use the 3 mobile phase concept for small molecule, for example, in pharma, and we look at impurity. Usually to develop method for impurities, it takes a lot of time because you need to develop the method for the main ingredients, but you need also to be able to detect impurities. And usually it takes a lot of time. If you don't want to lose time, you can use the CDS, the tree mobile phase concept. Here, we have two examples. We have the acyclovir and the spironolactone. And you see that using the medium polar developing solvent, the MPDS, we are able to separate the spironolactone from its impurity C. So without the optimization, we are able to separate them. Same thing for the acyclovir. Using the HPDS, we are able to separate the two compounds. So there is no need of optimization or method development when you are using the tree mobile phase concept. Now, if you think that all your samples are tested with the tree mobile phase concept, so they are unrelated, you can analyze pharma product, ingredients, you can analyze plant, botanical, finished product, everything. You will test them using standardized HPTLC with the tree mobile phase concept, using derivatization, and you will save everything in a database. When you do that, imagine what you can do with this database. We have created this database with different compounds. We have done a database of more than 200 reference substances. And from that, you have here the list. You have the RF value obtained in the 3 different mobile phase. And we have the SDF. SDF is the structural data files. From the SDF, we have properties. We have molecular properties. And then from that, what we are doing, we create a model that links the RF to the molecular properties. What does it mean? Before, when you had one zone that was unknown, what were you doing? You were just scratching the zone, dilute, filter, and then analyze this zone in MS, for example, or NMR. When you had the instrument, what you were doing with the interface was just going onto the zone, have a HPLC pump connected, extracting the zone from the plate, and connected to the MS. And you had the MS, for example. Now, what you can do is using your database. You have the query. Your query will be the 3 RF value that you obtain for this unknown zone. You will enter the query into the model that we have created, and the model will search in your database, and then will give you the potential matches. It will give you which compounds give you exactly this RF value. And then you will have information already. You will have a prediction. So in that case, you don't need to buy thousands of standards. You will just purchase one standard that this model is giving you. And you will verify that. This, it's the case of the machine learning. But now, what if you don't want to test everything? If I'm going back to the spironolactone example, here we have the RF value of the spironolactone, the main ingredients, with the 3 mobile phase. And we have this impurity. So we know that there is an impurity A. We have the name, so it means that we have the SDF. It means also that we have the molecular properties. But what we want to know is what are the RF value in the 3 mobile phase. You don't want to do all the tests. You don't have the time. So you want to predict your RF. So for that, we have a model. And the model is predicting the RF value now. So we have here the prediction. We have the compound A, impurities, 0.073 with the low polar, 0.63 for the medium and 0.97 for the high. We did the test. We had these impurities, and here are the real value. And you see that the predictions are not far. So we can really predict RF value now. So it means that you don't need to do all the testing all the time. Using the 3 mobile phase concept gives you more power and let you gain some time now. What is next? Here we talk about RF value, but HPTLC, I told you, is not just RF value. There is multiple detection. For each zone, we have UV spectra. For each zone, we have MS. For each zone, we can also have toxicity. So what is next is multi-dimensional hyperspectral of HPTLC. We can combine all that. So to conclude, what we are seeing today is that botanical analysis is becoming more complex. And at the same time, we are expected to make faster and more reliable decisions. What HPTLC offers is a way to simplify that complexity. It allows us to see the full chemical picture, to detect risk, and to decide with confidence. And importantly, this is not something for the future. It is already being used, and it is becoming more powerful. We are moving from analysis to decision, and now to prediction. The question for you is no longer whether HPTLC works, but whether you are ready to use it. Thank you.
Okay, very insightful presentation. Thank you, Dr. Jo. Now, time is for Q&A session. Participants who join here, feel free to raise your hand. Okay, thank you.
Selamat pagi, Bu Profesor. Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi. Good morning, Dr. Tien. Bonjour. Vous parlez anglais?
Yeah, I speak French, English, Vietnamese.
Oui, okay. Speak English better, ya. My name is Irfat. I run the lab. Name is Laboratorium Service International Indonesia. Lab testing, just recently. I also representing of the AB3O, which is Association for Producers of Biopharmaceutical and Raw Material Pharmaceutical and Food. In my life, actually I imported this TLC. I worked for Merck before, for the last 8-7. So I know TLC very well. I'm not chemist, I'm not pharmacist. But I have one pharmacist which is very, very, very experienced. 20 years, I think, I trained him overseas to learn about TLC on SPLC. Look at your presentation, actually, I agree with you that this is not misidentification, this is more on the adultery, okay? Especially sildenafil. Okay, I look at all the gentlemen in here. Yeah. If not mistaken, ya Pak, we need sildenafil, ya Pak. So, my very simple question, in regard to your method of analysis, yes, I agree with you. This is the simple way, yeah, to identify this adultery, yeah, either from herbal, which is in this country is very, very common, yeah, herbal, yeah, and we are all wants to be strong, especially after the gym, yeah. And my question is, how many minutes, how many days that we can identify this using the HPTLC, high porosphere thin layer chromatography, right? That's the name, right? I used to sell it very cheap, yeah, and promote it to all the lab. Just use this one to identify the adultery, yeah, from the customer, from food, from pharma, samples, everything. But yes and no. Sometimes there is a question about the integrity of the data. Yeah. But now I strongly believe this is the future. Yeah, to preventive, not to do correction. Here in this country, we use the kappa, corrective action, preventive action. I prefer to use preventive action rather than corrective action. Preventive is much better than the corrective one. So if we can do this one quick, yeah, especially for the FDA Indonesia team, usually in the many places across our countries, or to the lab, that we can receive it, the samples of these herbals, food, that we do not know the content exactly that you just mentioned as a fingerprint. I believe this will help us to avoid more and more disease in this country. Thank you.
Want to answer?
So, for example, the method that we have developed for the sildenafil, this method was done in the project joined with the FDA and the USP. And they asked us to develop a fast method. And the method is really fast. Developing a solvent and developing time was only 10 minutes. And then if you do the saturation, at the end, when you apply, you can apply at least 15 samples on one plate, minimum. So you can change the configuration, the layout of the plate to apply more. But if you have at least 15 samples on one plate, and then you do the application, the development, there is no need of derivatization. With the sildenafil, you can detect before. It's less than 30 minutes to have at least 15 samples. And you can detect not only sildenafil, you can detect all the analogs that are also known now on the market. So it was really easy, and we did this project on not just tablets. We did this project on gum, on syrup, on chocolate, on many gel, different things. And everything can be applied on the plate without a big sample preparation. So this was a really good tools for the USP, and that's why it's part of the USP now. There is the General Chapter 2251, used for undeclared drug. So sildenafil is part of that. And then the second project was for the weight loss product. The weight loss product, as we know that they are adulterated with sibutramine, but actually they find out that it's not only sibutramine. They are adding more compounds inside. Antidepressant, that you feel good. They add also some caffeine, that you feel more energy. They add, we don't understand why, but they also add sildenafil inside now. So I don't know the relation between that and the weight loss, but if you feel better, that's why you are using it. So that's why now there is another project where we did with the FDA for that. Same thing, it has to be fast. We have to be able to detect all the drugs now. So HPTLC is not just the comparison that you had before with TLC. It's really a good tool that the regulation are using now.
Thank you very much, Dr. Tien. This is very helpful. In our neighbor country, vape, electrical vape is already prohibited. In some country, cannabis is also can be used, not in this country. Okay, our generation of us now using this antidepressant or sildenafil or any drugs that in related with the vape. This is very, very scary, really. Yeah, they want to be relaxed. They want to be, So I hope we can work more detail later on how do the hands-on together in the lab, how to control this one. And I do hope the FDA Indonesia can issue the regulation to do the method of analysis, the SOP and everything to avoid this kind of the problem, which is already 10 minutes up to 30 minutes for 15 samples. So this is very quick, right? Again, thank you very much.
Thank you. Thank you. Are there any questions? Okay.
Yep. Thank you for a good presentation, Dr. Tien. Yeah, my name is Indra from Equilab International. We are one of the external party the external laboratory in Indonesia. So actually, I have never using this HPTLC before, just a normal TLC that we have been performed in our laboratory. Just curious with some of your presentations. The first regarding the system suitability test, the system suitability for this HPTLC. You mentioned that you using the general system suitability, and you mentioned for about 8 molecules. So in the daily of the HPTLC operations, so meaning that if we are not running in the mobile phase that have been validated, so can we also claim that our data with our mobile phase, specific mobile phase, for example, as long the sequence same with your same with the validated method, so we can claim directly that it's already proper with our analysis or not. And then the second regarding the molecules that lacking of the UV absorption, yeah, for the derivatization, what kind of the critical parameters for the molecules that need derivatization after the post-chromatographic process? Yeah, thank you.
Can answer the question.
So for your first question about the system stability test, so just I forget to mention during the presentation that the UHM was now officially recognized by the European Pharmacopoeia. So it means that the European Pharmacopoeia will always use the UHM on each plate in each method now. So it means that when you want to be compliant to Pharmacopoeia, you have in all the case to use SST. So or it's the new SST with the UHM like the European Pharmacopoeia, or it's the old SST that they had. But to be compliant, you have to show that you did everything in a good way. So SST is actually not something just in HPTLC. SST is also used in HPLC and in GC. So it's like when you are, it's what you call the control sample, control standard. And the control standard all the time has to be the same. Here is the same thing for the SST. Now, when you follow regulation, you will have to do SST. It's mandatory. So you have to do that. And to simplify this, we have this UHM. This was really to simplify everything. So if you are trying to reproduce or transfer one of the methods from the Pharmacopoeia to your lab, you have to show that you obtain the same sequence of zone as them. And when you show that, then you prove that you can use the data for the evaluation after. The second question was?
For the derivatization.
Derivatization. So most of the time, derivatization in HPTLC, we use specific reagent. So we have, for example, natural product for the flavonoid, anisaldehyde reagent for the triterpene, nidrine for the carbohydrate, or aniline for the amino acid. So we have specific reagent when we need to derivatize, when we don't detect them. We are working on it now, so we finished to work on it, and we will publish the article soon, is a universal derivatization reagent. So one derivatization that will be able to detect everything. So we are writing now the article, so we will submit soon, so you will see it soon. So like that, we will use only one derivatization, and we will combine with different detection mode. The before derivatization, after derivatization, and using the scanning densitometry. And like this, we will have a full vision of what you have in your sample.
Yep. Okay. Maybe can I follow up my first question? Yeah, for the system stability. So for the system stability that is already validated with the sum of the mobile phase, I mean the specific mobile phases for the system stability. How about if we have a new mobile phase that not validated before? So for the plate, for the SST mix itself, so we have to validate first with the validated mobile phase, or For the new mobile phase, we have also already, we have to validate first before we doing the analysis for our routine.
Yeah, exactly. So when we work in the pharmacopoeia context, usually when we develop the method, we are doing the validation after. And during the validation, you are doing at least 5 plates, different days, different analysts, different time of the day, and then you are comparing the error value. And from the 5 plates, you are calculating the deviation that you will accept. This deviation is where you put them in the method after. So if you use the pharmacopoeia method, they are already validated. They have been tested in different labs. If you, in your case, in your lab, you are developing new methods, then you do the same work. You will check the validation, you will check the reproducibility of your plate using the SST, and you will have acceptance criteria for your method. It's exactly the same process.
Okay, clear.
Okay, thank you for the question. Now we move to the online question. Maybe we can see the question for the online participants. Can we see the online question or have to? That's the first question from the online participant. How can we ensure that the HPTLC method can be used to detect the illicit products? Since we have to maintain the effectiveness of the analytical source, can it be used to decide the result without any other method for confirmation?
So usually the method that we publish when we work with regulation are validated. So they can be used for detection of illicit products directly. So most of the time when we want to have a confirmation method, in all the case, the confirmation method it's done by MS or NMR. So in HPTLC, you will screen multiple samples, you will detect zone, and those zones you will have an identity. But the confirmation after, usually you can extract from the plate directly and send to the MS. And then like this, you will have the confirmation. So it's usually how we proceed with illicit product.
Okay. We move to the second question. Going forward, is SPTLC expected to replace SPLC, or will both methods remain in use for laboratory analysis? Then which one of 2 instruments would you recommend more for our lab's current setup? Thank you. From Bipom Mamuju.
So HPTLC will not replace HPLC. So we don't want to compete with HPLC. We have different goals. HPTLC is used for identification. It's the first technique you will use for identity. And that's why it's always the identification is done by HPTLC in most of the pharmacopoeia. HPLC has, of course, a better resolution, has it's more sensible in some case. So HPLC will stay for quantification assay of some ingredients. And we complete all the data together. So we do the identity with HPLC, we do the quantification with HPLC. In some case now, in the European pharmacopoeia, we have, when HPLC is too difficult, when the method in quantification by HPLC is too difficult or too expensive, then they ask if we can do it in HPTLC. So this is the only thing where, in some case, we replace HPLC. But you have to know that HPLC, it's here for identification. It's a really good screening tool. But we are not competing with HPLC. It's not the goal. We work together for the identity of the product and the safety of the product.
Okay, thank you. And then the third question. Okay. Regarding SPTLC standardization, are there the chromatograms database as a reference for all the herbal medicine materials? This is to verify that the medicinal plants meet the required standard. From Sri Slamet from PT Auto.
So the HPTLC data, so the fingerprint that you obtain could be used as reference material. So until now, in the European Pharmacopoeia, all the chromatograms are in the knowledge database. So you can access online, and the knowledge database is free. There is always an example of the result. The European Pharmacopoeia has a table to describe the fingerprint. There is no picture in the monograph. The USP until now is the same. There is no picture and no standard. There is only in the dietary, the supplement information, you have some picture inside. But with the evolution of the digitalization now, the question is, will come. Are we using the fingerprint as digital reference? So it's still an open question because it's asking many different parameters. But I know that in different pharmacopoeia, they are working on it. They are trying to see how we can use HPTLC fingerprint as digital standard. So it's something that will come.
Okay, thank you. And then for the fourth question. Oh, a lot of questions. There's the two questions. With rapid advancement in AI and automation, how does the SPTLC Association envision the integration of AI into densitometric reading and fingerprinting chromatogram interpretation, specifically for the detecting adulteration in feed raw materials in the future? You answer first question first?
Yeah.
Okay.
So for the integration of AI tool and chemometrics into regulation, so I don't know if you are aware that the USP has already a general chapter on chemometrics. So it's already described how to use chemometrics in the regulation part. The only problem with this general chapter is that it's not really clear how to apply this chemometrics in routine analysis. And that's why right now we are working on a project together with USP to show an example how in real life we are using that, how we can integrate all the chemometrics into the regulation in quality control. So this is what we are doing right now. For your question in the feed field, the feed is more complicated because feed is many lipids, so it's really difficult. But the difficulty is just the sample preparation. It's not the analytical technique that is used, the chromatography that is used after. It's more the sample preparation that is difficult.
You also answer the second question. Okay.
For your second question about many feed laboratories in developing countries often find themselves caught between using kits, ELISA, and all that. So, HPTLC started to also go into the direction of biotesting. So, I mentioned in the presentation, we start to do some toxicology on the plate directly. There is more and more methods on biotesting now, with enzymatic reaction directly on the plate. Some labs are using the HPTLC to see toxic compound coming from packaging into the food directly. So there is different things, and HPTLC start to be positioned into this field also. And apparently from what the lab are saying, because I'm not biologist, so I don't really know, but from what they are saying is that HPTLC allowed them to have more precise result and can go down into the detection level of toxic compounds.
Okay, thank you. Okay, move for fifth question. Is finished or there is next question? It's finished. Okay. Still there is a question? Okay.
Sorry, there are two more questions.
Two more questions. Okay, this is the question. In your opinion, which provides a more reliable assessment of herbal quality, quantitative analysis of marker compounds or chromatographic fingerprinting? Do you think a single marker compound is sufficient for quality control, or should multiple marker compound be used to better represent the overall quality of the herbal product?
So to answer this question, I will give you an example. We go back to the ginkgo example. The USP came many years before with one monograph where they were saying that for the ginkgo, we need to do the quantification of flavonoid based on the rutin. So then what the people are doing outside, they add rutin. And then they calculate the quantity of the rutin inside. So all the other ingredients were below, but the rutin was good. Then they did the same thing on quercetin, because quercetin is also a flavonoid. So they also add quercetin. So if you look at individual ingredients, single marker, it's easy to fake. It's easy to add them in the product and to reach the limit that is asked by the regulation. It's what we are seeing more and more. So if you focus and stay focused on specific marker, the people outside, the one that want to adulterate your product, they will just add them. It's easy to do. And it's, in some cases, way less expensive than buying the real product. So for me, staying with the single marker compounds is not the right direction. Doing quantification on single marker compound when it's a toxic ingredient, yes, you have to know the limit. But for the identity, focusing in one single marker, it's not good. We have to look at the entire profile. Be sure that all the ingredients are here. And you know that when you use a product, it's not just for one single compound. It's the synergy between all the compounds that make the product effective in botanical. So you have to look that the entire product is of the good quality, intense, and not just diluted.
Okay, maybe for the last question. Okay. What quality control practices do you recommend to ensure long-term reproducibility of SPTLC fingerprint libraries? And how should laboratories maintain consistency when multiple analyses perform SPTLC fingerprint interpretation? Do you foresee artificial intelligence eventually assisting or even automating SPTLC fingerprint interpretation?
So to be sure that you have reproducible HPTLC fingerprint, you need to be standardized. You need to mention all the parameters that you are using. So for that, there is, in the association, there is an SOP that you can download for free. And the SOP is explaining you how to perform HPTLC in a reproducible way. When you have this reproducibility, then it's where you can use machine learning, data mining, and artificial intelligence. HPTLC will go into that direction in all the case. We have picture. From the picture, we have information. And from this now, using all the tools that you have in artificial intelligence, then we will for sure be strong into that. We will need to, we have to go out from this interpretation that only the user had before, an experienced user. So now with all the tools that we have, we will be able to have directly something telling you it's compliance or not with confidence.
Okay, thank you for all of the question and all of the answer. Dr.
Okay,
Tien.
sorry for interrupt, Professor Dr. Ida. There is one last question again from
okay.
the online
There is a last
participant.
question.
Yeah.
Okay. The next question, the last question, I think. What competency should analytical scientists develop to maximize the potential of modern SPTLC?
HPTLC is simple as technique. So the main competence that one scientist should have when he wants to do HPTLC is really to be rigorous and work in the standardized way. Then after, it's depending if you want to develop method, then you need to have some basis in analytical chemistry and chromatography, of course. And yeah, otherwise, I would say HPTLC is actually quite simple to use.
Okay, thank you. Before we close this session, would you mind to explain about the SPTLC Association and also Indonesian chapter?
So the HPTLC Association was created in 2012 in Switzerland. And it's now we have more than 270 members of different parts of the world. We have a local chapter in the HPTLC Association. We have, for example, the US, the North American chapter. We have the German chapter, the French chapter, and we have Chinese chapter. So each local chapter is working individually to promote and propagate the knowledge in HPTLC. Here we have the Indonesian chapter, which is really active because it's the chapter that is the more active that we have. You have many members here, and you are developing a lot of methods, workshop, helping user into having standardized and reproducible results. So the association is working a lot in collaboration between different universities, different industries. If, for example, people want to validate their method, but they have only one lab, they can ask to other members if they would be able to test their method for the validation. So it's what we mentioned previously. Collaboration is essential if you want to progress. And the HPTLC Association is done for that, for collaboration and work all together to promote and see the advantages of HPTLC.
Okay, thank you. Maybe for addition, there is a Indonesian chapter association, SPTLC association here. If you want to collaboration, you can contact us, and we make a collaboration for the development of the method. Thank you, Dr. Do, for the nice and valuable presentation. And I hope now everybody know about how valuable, how great, how powerful of the SPTLC method. I think thank you for joining with us in this session. I finish the session and thank you for the attention. Thank you.
Thank you.
Thank you, Dr. Tiendo, for your excellent presentation, and thank you for Professor Dr. Apoteker Ida Kusumawati, MSc, for expertly moderating the session. And may I kindly invite you to remain on the stage. As a token of our sincere appreciation for the valuable insight and expertise shared during this session, we will now proceed with awarding of a commemorative plaque and certificate to our distinguished speaker and moderator. And may we respectfully invite Ibu Mimin Jiwa Winanti SSI Apoteker to come forward and present the plaque and certificate to our speaker and moderator. Thank you.
Ladies and gentlemen, please join me in giving them a well-deserved round of applause. Okay, thank you for the valuable knowledge and perspectives shared with us today. We truly appreciate for your contribution. May we kindly invite Ibu Mimin Jiwawinanti, Dr. Tiendo, and Professor Dr. Apoteker Ida to return to your seat. Thank you. Okay, thank you, ladies and gentlemen. Innovation begins with curiosity, while sustainability begins with responsibility. This discussion this morning have demonstrated how laboratories continue to evolve to meet future challenges. Ladies and gentlemen, we will now adjourn for the lunch break. But before that, we have one exciting announcement. At the end of today's seminar, we will hold a lucky door prize draw through a spinning wheel to select our lucky winners. So make sure to stay with us until the closing session this evening, as you might be one of our lucky winners.
Okay, ladies and gentlemen, now participants are kindly invited to enjoy your lunch at the designated outdoor dining area located beside the doorstep entrance. And our distinguished VIP guests are respectfully invited to enjoy lunch in the VIP room. Our committee members will available to assist and direct you to the respective dining areas. Enjoy your lunch, and we look forward to welcoming you back at the afternoon session at 1 p.m. And please be returned to the seminar hall 15 minutes before the next session begins. Thank you.
Okay, now we end this boarding session. Wassalamualaikum warahmatullahi wabarakatuh.
Baik, Bapak dan Ibu sekalian, untuk lunch atau makan siang diharapkan untuk tidak makan di ruangan, sehingga dimohon untuk Bapak dan Ibu untuk makan di tempat yang telah disediakan oleh panitia untuk makan siang. Terima kasih.
Okay, Assalamualaikum warahmatullahi wabarakatuh. Ladies and gentlemen, thank you for coming back to the seminar hall. And this session 2 of the seminar will commence shortly on 1 p.m. So we would like to invite you to be seated in the front. Silakan Bapak dan Ibu yang terhormat, terima kasih telah kembali ke ruang seminar ini. Karena sesi 2 seminar akan segera dimulai pada pukul 13.00, kami undang Bapak dan Ibu sekalian untuk mengisi tempat di depan. Terima kasih.
Recording in progress.
Untuk Bapak dan Ibu yang masih berada di luar ruangan, diharapkan untuk segera memasuki ruangan karena acara akan segera dimulai. Terima kasih.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. Welcome back, and we hope you had an enjoyable lunch break. Okay, so we begin the second session of this international seminar. As laboratories continue to evolve, digital transformation has become one of the key drivers of innovation, enabling laboratories to become more connected, efficient, and data-driven. The future laboratory is no longer defined solely by advanced instruments, but also by its ability to harness digital technologies and support informed decision-making. We shall now proceed to session 2, entitled Digital Transformation, Automatization, Sample Preparation. This session will be presented by Christia, the chromatography and mass spectrometry specialist.
This session will explore how emerging digital technologies are reshaping laboratory operation and supporting the development of smarter and more resilient laboratories. Without further ado, I would like to invite our moderator, Bapak Dr. Andes Tepi Usia, Apoteker MPhil PhD, to lead the session. To our speakers and moderator, the floor is yours.
Okay, good afternoon, everybody. So, ladies and gentlemen, and welcome back to the session 2 of today international seminar. I hope you all enjoy the previous session. And ready for another insightful discussion for the section 2. So, as we look toward for the future of laboratory operation, and also efficiency, accuracy, and scalability, have become paramount. And this brings us perfectly to the theme of our session. Second session. This is the digital transformation.
Yes.
About the automation and sample preparation. And so I think Mr. Chris. So I call you Mr. Chris.
Yes.
Okay. Thank you for attending this seminar. So this is will guide us, especially Mr. Chris is the application manager, for the chromatography and mass spectrometry division at Thermo Fisher Scientific.
Yes.
Thank you. So I think please we have about 30 minutes.
Sure.
For presentation.
Okay.
And maybe about 15 minutes for the discussion.
Okay.
I think if you have any question, you can write in the chat box for the online attendant. And for the in the here, you can write, and after the presentation, we can discuss about what is the point of this our theme.
Okay.
Okay, please, Mr. Chris.
Okay.
Time is yours.
Terima kasih, Bapak. So, good afternoon, everyone. Thanks for joining the session. So, can you see the slide now? No?
Maybe the host, you operate from here?
Yes.
Okay.
I attached it to HDMI. Maybe we need some technical assistance. I want to. Okay. Okay, great. Good afternoon, everyone. My name is Chris. Thanks for inviting me for this session. I think it's a great honor to be invited in this seminar. So on my talk this morning, we heard about driving sustainability. So what is actually sustainability? So when we talk about laboratory works, so it's not when we talk about sustainability, it's not always about how to reduce waste. So it's about how we can achieve more quality result with the less resources that we have. So now, if we look into the modern laboratory challenge, so now this is the challenges that we are facing. So be it you are in pharmaceutical lab, research academic, even in petrochemical industrial, you face all these challenges. So if you can see, we're trying to do more analysis with fewer resources. So it means that we have less people in the lab, but we want to achieve more. We want to be a bit comprehensive in our workflow analysis. So about modern laboratory, we always need to equip with advanced technology. So we want the system to be more selective and more sensitive. But we want quality result. So when we deal with data, okay, where we want to keep our data, do we want to keep as previously what we are doing? We use pen drive, we use external hard disks to do a housekeeping. Or you want to be able to share your result with your research peer. Maybe for your colleague, not only in Jakarta, maybe in Sulawesi, Samarinda, how you can effectively share the result. So this have to do with the cloud-based data export. How you can even consolidate your data, your methodology, and you can actually share with them with a cloud base. Okay? We want to make sure our data is kept confidential. So when we're dealing with a certain new initiative, new project, how we want to keep the data confidential only to a certain group of researchers. So this, we have to always increase our awareness and to avoid cyber attack. Regulation is ever changing. Okay, so this morning we talked about regulation. Okay, we talked about certain analysis we yet to have threshold, but we have analysis maybe like PFAS. We already know the concentration or the genotoxic impurity from certain pharmaceutical products. So all these thresholds, they're not going to stay there. It will go down to a lower concentration to protect consumer like us. So we want a threshold which is comfortable, not only for us, for younger children, and even for baby. So if you talk about pesticide analysis, I think this analysis is quite comprehensive. Everyone know about pesticide. So what is the pesticide threshold in food product, in baby food? In baby food, the concentration is down to 1 ppb. But normal food that we consume, vegetable, fruits, maybe it's a range of 10 ppb to 20 ppb. In water, it have to be at lower concentration because all these pesticides, they are actually leaching from our agriculture field to the water resources. So different product that we are dealing will require different requirement and the concentration. So this is why the regulation is always updating and it will become more stringent. So this, for us that work in the lab, is a requirement for us to always look ahead. When we are getting a sustainable and modernized method, don't always look at current situation. We should always look forward about 3 to 5 years ago, anticipate the changes that required. So of course, it's always important to give a very short analysis so that we can address public in a fast manner. So this is why when you deal with pharmaceutical product, food safety, we always need a fast response team. So all these 6 pillars will make up to modern laboratory challenge. Okay? If we talk about method, so why method require constant update? So if you look at this, the one on the left side, okay? So this is one method. So this method required solvent, reagent to prepare a sample. So we want to minimize the solvent usage. And now we are more concerned about the operator health. We don't want them to consistently expose to this chemical. So this is why we tend to find a more modern method to minimize the usage of excessive solvent. Solvent like xylene, kerosene, we try to minimize, okay? And we try to minimize waste disposal. Time consumed, how long you need, how much time you need to prepare a sample. Is it 3 days? Is it something that acceptable to us? Or we just want something that can be prepared in half an hour? So remember the earlier talk that we have before lunch, we talked about fast analysis. So this is what we are focusing. And most important, after all this concern, we don't want any ambiguity in our result. We want the result to be consistent and accurate. So just a simple example for liquid extraction. I think during bachelor time, everyone deal with this liquid extraction. So we will have a huge funnel, maybe like 1 liter. We will pour about 500 ml of sample, and we will add 300 ml of solvent, mostly like hexane or dichloromethane. So we will have this to do extraction. So this is actually the step required for manual liquid extraction. After extraction, we will do concentration. So we will blow dry the solvent and concentrate our sample. But do you notice that from this process, we use a lot of solvent, a lot of man-hour. Man-hour means that we need physical person to deal with the solvent, and there's no automation. So the time consumed, the solvent that we consume, and also the sample that require. So what if today someone told you that, well, you don't have to do all these manual steps. You can leave it to auto sampler to do automation. With automation, it saves your time. The result will be more consistent. It utilizes less sample and less solvent. So sounds a good idea? So it have to do with reduction of the solvent consumption. So if you can minimize the solvent consumption, it's actually cleaner or better for the environment. You don't have to deal with so much wastes, and you don't need someone to collect waste so frequent. So this is why, whenever possible, we would tend to go for automation, and we scale down the solvent consumption. After we all do this, we will never how do I phrase it? We will never reduce the quality of our data, because the technology now is more sensitive and is able to detect at lower concentration. So we used to detect at microgram, so now we are doing at nanogram and even picogram. So some technology can even do at femtogram. So with the advancement of the technology like chromatography and mass spec, it enables us to reduce the consumption of sample. So for example, if you need to use about 200 ml of solvent per sample. 5 samples will use up a liter. So what if I can reduce this to only 2 ml? We use less solvent, and a bottle of 4-liter solvent will be able to accommodate 500 samples. So this is the automation that currently most of the lab that doing volatile organic sample in water are considering. Okay? With the usage of less solvent, it will imply that we will use less sample. Your colleague that goes to the field, go to the on-site to collect sample, they will collect in a valve rather than in a huge bottle. So it involves transportation, it involves storage. Okay? So all these improved logistics will actually speed up the space and time required to transfer all the sample to field to your centralized lab. So this is the example when we deal with VOC and SVOC in drinking water. It can be in groundwater, in river water. So when you need to transport the sample, last time we used to transport in 2 liter. 1 liter for our analysis, the other 1 liter for backup, mean for storage. In case something happen, you need to repeat the analysis. But now we will just store in a 40 ml sample tube. Okay, that's the scale down from 2 liter to 40 ml. And just imagine that the solvent required to process that. So it will even scale down. So this is why when we talk about sustainability, you need to start thinking from the sample preparation to the analytical workflow, and later you will think about software. How the software will make your data more reliable and how you can upload them to cloud-based. So why sample prep is important? Because it helps us to prepare the sample in the adequate condition so that it will able to detect by our methodology. So some will detect PPT level and some will be at PPB level. Okay? And we also need to reduce background. Okay? So if you heard about nitrosamine, so nitrosamine in pharmaceutical, I think is a common terminology. So, but nitrosamine also present in our wastewater because of dumpage. Okay, so we may release some nitrosamine from cosmetic, from our product to our wastewater. And when the wastewater get recycled, it will form nitrosamine. So nitrosamine, if we were to detect in our wastewater, we will need GCMS, MS for detection. So in order to do that, we need to reduce the background. So this is why you need to perform sample preparation, and we need to enhance the signal. So enhancing the signal will mean that I will need to consolidate or concentrate the sample, and so that it will able to be detected at least 2 nanograms. This is the current requirement for most of the lab, I would say water lab, okay? And enable lab to meet method regulation. So currently the method is about 2 nanogram, and we will ensure that we were able to reach lower than 2 nanogram. Okay? So earlier example is about water. How about when you have solid sample? How you going to prepare it? So we don't want a technology that require us to use multiple systems. We want a single system that able to perform automated extraction, pressurize, and automate. So why this is important? Because it reduces solvent. It able to perform automated cleanup, okay? and most important, to perform evaporation. So all these three is equally important, and it enables us to prepare the sample in the final product that we required. Okay? So what's next? So why do we need such technology? Can't we just use a normal extraction? Because the pressurized extractor will use less solvent and is able to heat up the sample pressurized with nitrogen gas. So when all these three scenarios add up, it will improve the extraction efficiency, improve your sample recovery. So this is why these are the new method for most solid extraction. Okay? And started, you will see that some regulation alignment that no longer accept simple extraction. So they want this pressurized extraction, and for example, PFAS in sample preparation. So how you're able to validate the testing and validate with the EPA method. Okay? Faster processing time. So when we talk about faster processing time, we are not going to prepare the sample one by one. Okay? Okay, so right now I have two diagrams. So you look at the lower diagram first. So this is the typical workflow that we are dealing with. So we will have certain automation, and we have some manual labor. Manual labor means that require technician or require chemists to perform sample transfer, extraction, adding solvent. So what if today we can minimize the manual labor and we pass all the job to automation? Okay, you just weigh the sample and you put in sample tube. The system will able to perform the extraction not one by one, but in a series of sample, maybe 4 samples in one go. So it will able to perform extraction for 4 samples in parallel. So that speed up your time, and at the same time, it utilize less solvent and a little gas. So it use less gases and less solvent. But with the temperature, when it pressurize it, the efficiency of extraction is increased. So this is why you were able to do more extraction with automation, less technician intervene. So it means that you just prepare your sample and just pass it to the extractor. So it will perform all the extraction, and you can save significant time so that you can look into other responsibility. You can do other analysis at the same time. So this is currently the environmental VOC and SVOC contamination workflow. So we are focusing more on automated liquid-liquid extraction. So I put a summary here. So you can read up that if I'm doing in-valve liquid-liquid extraction, I will only use about 10 ml of water, that's your sample, coupled with 2 ml of extraction solvent. So most of the time we will be using dichloromethane. And the system will able to do a vortex. So look at this little picture here. So this is where the autosampler will transfer your sample valve by magnetic valve cap, and it will able to do vortex, centrifuge, and followed by sample injection. So all this workflow is actually done automatically. So what you need to do is just prepare the sample in a 20 ml valve. So when we deal with pharmaceutical product, food product, you can use this dispersive micro liquid-liquid extraction. So it will only use 8 ml of your sample. So if your sample is solid, we will use 5 gram, and we will add acetonitrile. So acetonitrile is a common solvent that we are using now, and you will know why in later slide. Because we want a universal solvent that enable both liquid chromatography and gas chromatography analysis. So we are using universal sample extraction method. No longer like only ethyl acetate that's suitable for GC, or water that's suitable for LCMS. So this is why we are using acetonitrile. So if you are in petrochemical, so we will do this in-valve liquid extraction followed by cleanup for hydrocarbon index. So these are the 3 major improvements in the method that enhance data quality as well as reduce solvent consumption. Okay? So just now I mentioned sample preparation is one of the pillars that we should consider when talk about driving sustainability. So next is about challenges on analytical contamination analysis. Okay, so what are we facing? We're facing increasing sample throughput, analysis scope. So just a question. When you run the analysis, how many analytes you try to detect? Is it only one? Or you try to detect as much marker as possible? So we want to be a bit more comprehensive. So this is why we tend to have more analyte detection per method. So we may analyze 300 pesticide up to 800 pesticide per injection. Sorry. And in order to do so, we don't want to sacrifice the confidence in our result. Okay, we want to do it at a shorter runtime, comply to regulation, and able to generate the result at the shorter time possible. So if you look at this, you can identify your challenge, and there's a solution to resolve this. So if we have challenges, for example, for sample throughput, so one day maybe you need to run 50 samples, and your instrument only can capable to run 20 samples. So how are you going to resolve this challenge? So you can think about something called tandem LC. Okay, so tandem LC is actually a new, I would say, more adoption currently in the lab. And how we do that is a single footprint of HPLC or UHPLC, but with a dual injector. Okay, so you look at this schematic diagram. So this is an auto sampler. So your sample will place inside here, and the system, instead of just having one injector port, it will have 2. Okay, so you will know why we need 2 injector port later. I have 2 pumps, 2 column compartment, and lastly 2 detector. So these are LC detector. If eventually you want to connect with a MS detector, it's possible. So when we are dealing with this, I will have 2 methodology that can run simultaneously. I can run with the same method, or I can run with a different method. Okay? Each injector port will able to perform independent injection. They can do it simultaneously or sequentially. So imagine that you only have single channel. So what you need to do is analytical run followed by conditioning. So these are the common workflow that currently we are working with. So now I consolidate it to have 2 injector, and I will able to stack up the flow number 1 and number 2 at the same time. So when we are doing that, we are saving time and we can run more coverage analysis on the same system. The footprint doesn't change. Okay? So now we have two identical pumps. It will able to handle different solvent and deliver to the column chemistry that required. The two injection were able to run simultaneous analysis for completely different application, and it can also run similar application. It depends on what is your focus. Two columns, and lastly, you have two combination of the detector that required. So we are doing that improved method because, for example, if you look at Europe Environmental Agency, so they have a target. Their planning is to reduce any chemical and pesticide at least by 50% by the year of 2030. So in order to do so, they have to do education to the farmer to reduce less pesticide usage, to ensure that analytical method have the selectivity and sensitivity to detect the pesticide. So pesticide, we always use either GC or LC-MS to detect. So what we need for a future proof method is something called unique cross-confirmation. So what does cross-confirmation means? So pesticide, you have two methodology. So one is GC amiable. So pesticide like organochlorine, organophosphate. Conventionally, we use GC to detect. But LC is actually more coverage. It will able to detect more sample and I would say higher level of pesticide at lower concentration. So, but they are not contradicting. You should treat them as complement method. So in order to do so, I will have a multi-technique, but only one sample prep to prepare the sample. Then we will pass the same sample with acetonitrile into two instrumentation. Just for example, if I want to analyze around 700 analyte, about 50% can be detected both LC and GC. Then I have about 15% only detected by GC and the other 40% by LC. So it means that I cannot use a single method. I want to improve the coverage. I need to use two methodology. So this is why the same sample, we pass to two different methodology for GCMS-MS and LCMS-MS. So when we are doing that, we doesn't need to prepare the sample two times. Save our time and solvent. With a single sample, I was able to increase the result confidently by doing confirmation. So this is an example of one pesticide, okay? And you will see that it's able to detect using GC and LC. So what we need is actually the retention, matches our standard. We have the ion ratio check, and also the recovery. So you have two plot of different calibration curve, but the ion ratio and amount match the confirmed level, and this is why you can do an orthogonal confirmation. Okay? So with the analytical instrument alone is not sufficient. You need the help of software. So the software will actually help you to perform certain automation. And just now I saw something called AI integration. So this is a software enhancement that enable you to perform integration, peak integration and detection automatically. And how to do that? We need to have more predictive performance. So the software must be able to predict what is the typical sensitivity that we have. If it fails, what is going to do? So we will be able to re-inject the sample, recalculate the amount, or able to just stop the sequence, wait for user to perform next action. ICH method validation template. So this will significantly help the user because you doesn't need to create the method from scratch. So you can just import the method. E-workflow is something I mentioned earlier. So if I manage to create the method, I validate it. So now I want to share it with my colleague in other region. Okay, I'll just create the e-workflow where it consists of method, sequence template, and the result. So I'll just I'll just upload the data in cloud-based, and he or she in another region will able to download it, and they can just straight import to their instrument, and they can perform cross-validation. Okay? So the rest of the feature like sequence ready check, so it will able to check your solvent, check whether your system have the correct tools to start the analysis. Smart startup, it will perform leak check, it will perform equilibration. And some software will enable system check that you can start the sequence maybe later 7 in the evening. Okay? So all these feature is enable for us to increase our lab productivity. And with the system suitability check and intelligent run control, this is something that we can look into on later slide. So when I mentioned about e-workflow procedure earlier, so we are able to run the software right now enable us to perform sample run, analysis, and report generation in a few clicks. Okay, it ensures that we adapt to each lab SOP, and it reduces training. Training is one of the major discussion for each lab because you do have new user. Maybe the more experienced user, you will allocate them to a certain new project, and the new user required to learn about the software. So how we can adapt to it, and we ensure that the e-workflow procedure is a shorter and easier learning curve for the new user. And this enables us to perform system suitability check, ion ratio confirmation, and we can perform all reporting with a suitable calculation. And when we talk about automation, so this is something built-in system suitability, and it enables us to perform something like, I want to ensure my chromatography meet the lab requirement. So we have a test result, and we were able to perform maybe, for example, the repeatability of the peak area. Okay, you don't have to manually look into the result. The software will look into it automatically. So it will do QC, for example, every 20 sample. So it will make 3 injections and look into your internal standard repeatability. So you can set it less than 2%. So what if it fails? You have to make a decision. It failed. Maybe you can instruct the software to automate a blank injection, or you ask the software to perform the next QC injection. So all these procedure or automated feature can be designed by the end user themselves. Okay, you can instruct the software to do it, or you can let the software to decide. So this is the automation that the automatic decision is actually based on the result. And we want to do that because we want to avoid subjective decision by each operator. And we want to reduce solvent usage. So let's say you run a QC and it's not detected. Do you want the sequence to continue? So obviously it's not. So when the QC fail, I will do a follow-up action of re-inject. If after re-inject it still fail, I will just stop the sequence. So when we stop the sequence, the solvent flow rate will also stop. So this requires user to check whether the correct column chemistry is used or the QC sample is prepared properly. So all this data processing, AI, report review, and quick data review process is actually help us to increase operation efficiency. And you can see that you can do integration automatically, and the AI feature will help us to detect whether this is a true, genuine peak symmetry or is something from the baseline. So after we have all this sorted out, before we upload the data, so what you need to do, we have to make sure that the system network failure protection. Okay? So I do have more and more users asking about, can your software perform report upload to my server? So for example, I have customers that they need to upload their data every 30 minutes. So consistently, we have to make sure that the system is connected to our network and it enables the result to be uploaded. So to ensure this online 24/7 operation, we need a unique network failure protection. So this is part of the protection to protect our data from cyber attack. So we ensure that the system can be accessed 7 days a week, and the technology enable continuous operation. So we want to make sure that we doesn't lose any data if there's a power failure, okay? Or the system is able to continue reboot after power failure, and user can actually log in to the instrument to check the system status and edit the required running sequence. So if all these are taken care, lastly, we can upload our data. So archiving our data is very important. So we don't want a specific user to copy the data on their personal hard drive. Okay, so we want all the data to be consolidated, keep in encrypted form, upload to maybe a centralized cloud base. So next user that require it, whenever required, they can just download the data and they can reassess. Okay, so from the discussion that we have so far, be it from sample preparation to analytical equipment and to result software. So all we want to do is we want to improve our method to match with current modern lab requirement so that it enable us to run more analysis. And ultimately, I think everyone in this room only have one goal. We want to make sure our life is healthier. The product that we take, we have more confidence, and our environment is actually cleaner and safer. So with that, I will end my talks. Hope this discussion brings awareness to everyone, then we can continue to improve our lab efficiency.
Okay, thank you very much, Mr. Chris, for the incredible, insightful, and forward-looking presentation. Yeah. Okay, it is a fascination to see how digital automation is reshaping and the landscape of chromatography and mass spectrometry. Yeah. Okay, we will now open for floor for question and answer session. Yeah, to all our participants in here and also by Zoom. Yeah, maybe if you have a question, you can type in the box. Yeah, chat. Yeah, and in here maybe you can raise your hand if you have any question. Yeah, and maybe Mr. Chris will answer your question. Okay, I open for the session. Maybe we have 15 minutes, yeah, until 2 o'clock for the discussion. Okay, please, in the left side, if you have any question, yeah. I think this is important.
yes,
Okay, please, the man, please give the mic to the...
If you speak too fast, then I cannot.
Okay, please.
Okay, thank you for your presentation, Chris. My name is Ahatma. I'm from pharma industry. Maybe I have one question for you. If I'm not mistaken, you in your presentation you're telling about simultaneous preparation for LC and GC, right? So the one preparation for two instrument. I mean, can you tell us about the reference, maybe guideline from USP or another pharmacopoeia regarding to that process? Because it's really interesting for us. We can make our laboratory more efficient by implementing what you tell us before. That's my question. Thank you.
Okay. Yep.
Okay, thank you. Please, Mr.
Thanks
Chris.
for the question. I think this is a great question. For pharmaceutical product, I think you have a bit limitation because you need to comply to certain regulation, USP method or pharmacopoeia. Okay, the cross-contamination method that I mentioned earlier is actually more for food industry. Yeah, for pharmaceutical product, when I deal with nitrosamine, we try our best to use a universal extraction. But for certain product, let's say oral fruit compared to tablet, the excipient condition is different. It's a bit different. And in order to achieve, let's say, the good detection limit, very often we need to use different extraction method. And this is the challenge that we have. Everyone have the same challenge. So, but we try to utilize the same extraction method. So for myself, for any excipient that required to check impurity, I will normally use methanol to do a simple extraction and a spin down. I will do it in a small sample tube, about 5 ml. So I will take about 0.5 to 1 gram of sample, and I will add 3 ml of methanol. I do a quick spin down, then a vortex and quick spin down, and I transfer the supernatant to a 2 ml Appendorf tube, and I do a fast speed spin down again. Then I will do an injection. So that's the methodology that I adopt currently. So if you are following on certain regulation, you have to continue to follow that. Yeah, there's no shortcut with that. Yeah, at least for the time being. Okay.
Okay, thank you, Mr. Chris. So it means now not available for the pharma, still for food, yeah? So, but how to develop the method, for this equipment? You compare the traditional method, and you compare this is the same result like that?
Yeah. So we use a standalone method, existing method that we validated. Then we try to combine a few scope together. For example, a few analysis together, and we try to look into the recovery when we use a consolidated extraction method. and we reduce the solvent usage, and we check the sensitivity that we can achieve and the sample recovery. If all tick, then we will use the new method.
Okay. So until now, how many methods for the analysis using this technology?
Currently, I think veterinary drugs, antibiotics, PFAS, it can be from food, it can be from air. Then we have pesticide. Yeah, I think it's about 7 to 10 methods.
Oh, 10 methods. Yeah. All for food.
Environment and food.
Yeah. Including antibiotic in food, yeah?
Yeah, antibiotic.
Okay, thank you very much. So any question more, please?
Yes.
Thank you for time. My name is Bambang Guswandi. I'm from University of Jember. In your presentation, you also discussed regarding using 2-channel, right?
Yes.
Is it possible to using multi-channel because it might be more efficient and let's say many analytes can be analyzed at the same time? Thank you.
Yes. Yep. So the question is whether we can have more than 2 channels. So the answer is yes. We can do multiplexing. Okay. The highest I done before is 4 channel. Yep. So imagine that one analysis is about 8 minutes. If I stack 4 channels, I will able to get one result every minute. Yeah. So it's actually for higher throughput. Yeah. So we need fast response. Maybe it can be from manufacturing, it can be from any commercial lab that they require fast turn up. So when you pass 100 sample, and they will use only 1 hour to process it.
Okay, thank you, Mr. Chris. More question, please. Yeah, this is in the left side. Sini, Mas. Okay, please.
Yeah, thank you, Mr. Chris, for the good presentation. Yeah, Indra from the Equilab. Yeah, we are a contract research organization and also the commercial lab. So currently, we are also developing for the automation for the lab preparation, especially. So, for example, in the Equilab, we have several laboratories. One of them is the bioanalytical laboratory. We were working with the biological samples, like a plasma. So, averagely in a month, we can working with around 10,000 samples for the plasma. So, one of the challenges we face for this automation is, basically our sample preparation is very simple. For example, in the biological sample preparation, we only using 3 sample extraction. The protein precipitation, the simple one, and then the liquid. Another one is the more advanced, is a solid phase extraction.
Okay.
So most of our method is using the protein precipitation. It's very simple, like adding the methanol or acetonitrile, then speed, and then after that centrifuge, then separate it, then directly injected to the LC-MS system. One of our challenges is to standardize, for example, the apparatus or the equipment using for the sample prep, because we only sometimes using like a simple microcentrifuge, and it cannot be registered in the system, for example. So do you have any, or your organization or your company can accommodate, for example, to make a registration of the apparatus that using for the sample preparation? It's easy to be registered here in your system, or maybe another apparatus can be customized also in your system for very simple preparation like a protein precipitation. Yeah, I think that's. Thank you.
Okay, thanks for your question. Your sample is in well plate or in valve?
Our sample is still using the cryophile.
Okay, the valve.
The valve, correct.
Okay, yeah. So you have 2 options. One option is that with the automation, now the auto sampler will also come with centrifuge. Yeah, so it will able to do a quick spin after you add in methanol for protein precipitation.
Yeah, because we have to take the specific volume from our samples to another tubes or container, and after that we have to precipitate and also separate it.
Okay, okay. There's another way is using a robotic autosampler to perform the protein precipitation that you mentioned earlier. Yeah, maybe I will gather some detail, then I will share with you separately.
Yes, correct.
Because
Okay.
I have customer that they, I mean, not doing on protein precipitation only, they do on trypsin digestion, more for peptide mapping. Yeah, for biopharma. So that's for their specific research, but they are doing more on automation on their sample. Yeah, so I can share that with you.
Okay, thank you.
Okay, thank you,
think
Mr.
there's one
Chris.
more question.
One question. Okay.
Yeah, thank you. Thank you for your presentation, Sir Chris. My name Purba. I'm one of the consultant here. The questions, according to hospital labs and BPOM labs in Indonesia, what is the base of use of HRM that you have just presentations? And can the data be used directly for BPOM or KAN for audit legal proof? That's the question. Thank you.
What is your question? Only the question.
Yeah, the questions according for the for legal proof that using your what you
For
just
legal
presented.
proof of
Yes.
this method.
Yes.
Yeah, you use the legal proof
For legal.
for your method. You
Yes.
already try maybe about 10 method here. You already approved
Right.
and use apply to the agency to approve this method, like the
For-
USP or something like that.
Not
thank you.
towards legal approval, but it's more for in-house method adoption, whereby they improve on their sample throughputs. So the USP method, for example, US EPA, maybe they will, or AOAC, they may have their recommended method. So there's a default methodology, but the cost confirmation method that a lab implement is more like a method enhancement. So it still fulfill the sensitivity or method required detection limit as per default method. So it able to perform the required separation and detection limit, but it do it at a faster speed. Yeah. So I mean, you can request for approval, but most of the approval is actually from in-house, in-lab. So for example, they have their own team to perform proficiency tests. So they receive the sample, and at the same time, they use two methods to perform. One is their conventional method. The other one is the newly adopted method. So the new adopted method also perform or give a similar result, but at the faster pacing. So this is why they will adopt their new method for fast screening and confirmation. So
Yeah.
hope I answer your question.
Yeah, okay, thank you, Mr. Chris. So I think for the method, like in BPOM, yeah, we also developed the analysis method, yeah. Of course, for this equipment, yeah, Mr. Chris already said they have 10, yeah, 10 method already developed. So of course, if we want to buy this equipment, yeah, so we see what is the method and we compare with the result of our conventional method. It's the same, no significant difference, maybe we can approve. Yeah, so like that. So I think the thermovisor also still develop the method, yeah, using this equipment, yeah, and maybe more better like the mass, yeah, said, yeah. Maybe if you have the video, how to prepare the sample, yeah, and what kind of analysis you do, yeah, maybe we can more understand, yeah, and how many, because you said only need a little sample and also a little solvent, yeah, maybe compare 10 times, yeah, to the conventional method, yeah.
Certainly,
So I think this is very important because also now this is efficiency, yeah,
Efficiency,
for
yes.
the method, for the solvent and anything. And this is important, it's very interesting for laboratory, yeah. We can discuss more, but of course, if you have some link video, maybe you can share for the participant, yeah, and we can understand and we can discuss to use this method, yeah, in the future. So still any question? But I think this is already 2 o'clock, yeah. We close the session and we give applause for the Mr. Chris. Thank you very much for your insight presentation, yeah. I think this is we can continue discuss for this method, yeah.
Sure.
Okay, now I give back to the.
Okay, thank you, Mr. Chris, for your excellent presentation, and thank you, Bapak Tepi, for your expertly moderating the session. May I kindly invite you to remain on the stage as a token of our sincere appreciation for the valuable insight and expertise shared during this session. We will now proceed with awarding of a commemorative plaque and certificate to our distinguished speakers. May we respectfully invite Ibu Mimin Jiwawinanti SSI Apoteker to come forward and present the plaque and certificate to Mr. Chris. Okay, ladies and gentlemen, please join me in giving them a well-deserved round of applause. Okay, thank you once again, and may we kindly invite Mr. Chris to return to your seat. And for Dr. Andes Bapak Tepi Usia Apoteker MPhil PhD, we kindly invite you to remain on the stage as we proceed with the next presentation.
Okay, ladies and gentlemen, as laboratory continue to evolve, sustainability has become an essential pillar in shaping the future of laboratory operations. The laboratories of tomorrow are expected not only to deliver scientific excellence, but also to operate responsibly, efficiently, and with minimal environmental impact. We now proceed to session 3, entitled Green Laboratory. This session will be delivered virtually by our distinguished speaker, Mr. Aaron Desmond from My Green Lab USA, joining us online. The topic highlights the importance of integrating sustainability principles into laboratory practices and fostering environmentally responsible operations without compromising scientific quality and performance. And we are delighted to again having Bapak Dr. Andes Tepi Usia Apoteker MPhil PhD joining us once again as the moderator for this session. To our speaker and our moderator, the floor is yours.
Okay, thank you very much. I still continue, yeah, as the moderator for the session 3. Yeah. So for the session 3, we are talking about green laboratory. Yes, this is also important for us. Yeah, because in the session 2, we're talking about the how to efficiency for the solvent. Yeah, and anything. This is also related to the green laboratory. So now for the session 3, yeah, we have the honor to have Aaron Desmond. Yeah, from My Green Laboratory USA. Yeah. As the leading organization dedicated to transforming the scientific sector, yeah, And my green laboratory set the global standard for sustainability in science. Aaron will discussing how we can implement sustainable practices and drive this vital culture of change with our laboratory environment. So, Mr. Aaron, hello.
Yes.
How are you
Hello, thank you
today?
for having me. I'm
Yes,
very good.
thank
And
you
how
very
are
much
you?
to join with our international seminar in Indonesia. Yeah. So now
Yes, my
your
pleasure.
time. Yeah, I give the time about the 30 minutes for presentation. Yeah, about green laboratory, and after that we have the 15 minutes for discussion. Okay, please,
Fantastic.
Mr. Aaron.
So I'm going to move ahead and share my screen. So can I just confirm that you can see my screen? Oops, sorry. You can see this?
Okay. Yeah. Ok- Yeah. Okay.
Yes?
Your
Yes?
slide already appears. Please.
Fantastic. Thank you very much. Yeah, so just very briefly then, I do want to say thank you very much for having me join. I'm Aaron. I'm part of the business development team at My Green Lab. And as you've just very kindly introduced me, our organization is committed to the proliferation, the growth, and the support of a more sustainable environment and community in the scientific sector. And so My Green Lab is a non-profit that was founded 12 years ago now in a lab in California by a neuroscientist. And she really felt that her work was incredibly important, but really was demoralized by the impact that she had. And so that's where My Green Lab was founded. And really our mission is to build a global culture of sustainability in science. Depending on who you are in the scientific sector, then we have different ways to support you and help you contribute to that overall goal. And this is what we look like now. In just a little over 10 years, the organization has really grown exponentially and has become a global effort, as you can see, right the way through all 6 continents with scientific interest. There's real involvement here and a real community that we see continue to grow. So we're now working with over 5,000 labs globally in over 50 countries. We're actually closer to 58 countries, I believe now. Our lab ambassadors have actually exceeded 10,000 individuals. We're hoping to hit 12,000 soon. And that's just the tip of the iceberg. So it really has become a global effort. And I just want to take a step back and remind ourselves, why are we doing this? Why is sustainability in science so important? When we look at how resource intensive lab spaces are, which many of you will have firsthand experience with, it's really quite substantial. You're talking about 10 times more energy use per square foot than an office space, 4 times more water use. And the stat now is that between 2 and 3% of global plastic waste is coming from our labs. And when we think of that being made up of nitrile gloves, of pipette tips and test tubes, these small, seemingly insignificant pieces of plastic, it really adds up. And one thing that was said to me recently is that the global healthcare system, what we're really trying to achieve here is a healthier and safer population. But now that same global health system contributes about 5% to greenhouse gas emissions. So there's a real moral obligation here that really labs, sustainability, science we need to push to operate in a better way. And so our solution for labs is the My Green Lab certification. What we do is we work with the teams within the labs to upskill, empower, train, and really just bring awareness to the operations in a way that doesn't involve capital investment, but really looks at empowering your team, growing that culture of sustainability, and addressing where really your environmental impact is. And we do that by looking at these 14 key environmental impact areas. So these range from your purchasing styles, your water usage, chemical usage, but also the infrastructure energy, your plug load energy, and overall the culture. This is a program to change culture, ultimately, and it's been quite an impactful thing to see in labs. Not only are we reducing your environmental impact, but we're seeing labs create significant return on investment, real savings by going through these programs, these steps. And we can have a look at a couple of case studies in just a minute. In addition, though, what I think is often overlooked is that if we look at creating a more sustainable environment at the lab, we're actually going to create a safer environment also. Looking at swapping out hazardous chemicals for maybe something more environmentally efficient will in turn give your team a safer environment. And ultimately what we're talking about here is creating an engaged team, whether that's within your team or between the labs. And that's something that we always want to reflect in the organizations. So this is a case study that we ran a little over a year ago now with the Department of Agriculture in Colorado in the US. And this was just across 3 labs, 3 very standard labs that you could kind of see reflections of in many organizations. And again, what we looked at here was autoclave best practice, reduced usage of the fume hood, shutting the sash essentially, looking at their waste output and seeing where we could make adjustments, and of course energy management. And by the end of year, what they had seen is almost 200,000 kilowatt hours of energy saved and almost 500,000 gallons of water, which is really enormous. When we're talking about 3 labs here of an average team of maybe 15 individuals, those are really impressive savings to be seen, and it's what we see as standard across the industry. So this was a really committed team, and they really did quite well. This is another case study we did then with an industry partner of ours, GSK. Again, 3 labs, this time in Europe. But they saw huge savings, almost 180,000 pounds sterling, so about 250,000 USD in cost savings. And that's just 1 year. So there's really a compelling argument to be made here that, not only is sustainability going to, positively impact your environmental impact, but also, really saving you quite significant costings as well through the year. And so it was a 14x return on investment that they saw in just 1 year. So what does it look like then for a lab to go through the process? How do you achieve a My Green Lab certification and see the same sort of steps that same achievements we've seen other labs reach? So it typically takes about 6 months for a lab to go through the whole process, broken down into 3 very simple steps: your baseline, your implementation, and certification. What we want to do is first understand how your lab operates now, what it is that is relevant to you, and we want to engage the team through a survey. This is all done virtually on a software platform. And once we understand how your lab thinks, what it is you concentrate on, and where the real impact areas can be made, we move you into this implementation phase where we are feeding you logical, intuitive steps that you can put in place in your lab that are going to give you that impact. And ultimately, this is about behavioral change, making small tweaks to your operations that will give you those savings in time. And we have calculators that will allow you to see those savings generate as you move through the program. And of course, once you feel like you've done all you can, then you submit your data to us as a third-party verification. This is really well trusted. It has become the kind of global standard, as you mentioned earlier. And so we perform a risk-based assessment on the supporting documentation, and you receive your certification. It's a very similar process, really, to what you might see in an ISO certification. And so, you know, we want you to come back to recertify every 2 years, and ultimately to move forward. You know, this is about continuous improvement. The task at hand, the issue of sustainable science is an ever-evolving one. And so we need to keep moving forward. We need to make changes, make improvements, learn from them, and continue doing so in the years to come. And it's a very simple certification process. You know, it's a scored system. So what we want to do here is really not judge you on the amount of energy saved or the amount of water saved. What we're talking about here is, of the things that are relevant to your lab, how many of them have you engaged with? What can you show proof of having achieved in your lab? And ultimately, that's where your score comes from. And this is just an example of one of the calculators that we have that really does prove quite powerful. You know, in order to get the momentum behind really making changes in the lab, you have to prove the data is there. And especially so with scientists when you're talking about working with labs. So what we do is we have regionally specific estimates for your electricity cost, water, and waste. These are geolocated based on where your lab is. And then because we know, and we've worked with so many labs, you know, what changes a certain step might take, and what data you can generate from, say, temping up your -80 freezer to -70. We'll know the environmental impact of that. We know that it reduces your energy usage by about 27% for that model. And we can extrapolate that and tell you then, okay, over the coming year, you're looking at saving about 1,000 kilowatt hours just in that step. And in your region, 1,000 kilowatt hours of energy is worth X amount. And so very easily, we can tell you projected savings for your year to come that is going to move forward year after year, and is something you can build on. And not only is this a powerful tool to get backing from your finance team and from your management, but really to empower the individuals in the lab, your teams, to help them see that they can contribute to an overall goal, that what they do actually matters, is a really, really important and motivating factor, we find. And so is one of the kind of standout tools that this program has given to labs. And there's a real commitment here from the scientific community. We've seen it time after time that this is the way forward. So now we have been officially recognized by the US EPA and recommended by their purchasing program. If you want to be part of the UN Race to Zero, you need to have this certification. And in the academic scene, certainly in Europe and the UK, there's now requirements to have a certification like this in order to be eligible for grants. Grant bodies want to see that you're operating in a responsible and sustainable way, and so this is a very important thing to hold. And the list of organizations grows all the time. Whether it's universities, government agencies, NASA have recently joined, but also moving into the commercial sector as well. The names in bold here have committed to organizational-wide rollout. This is really becoming a standard product now to be delivered in labs, and is something that we should all aim towards. And as I say, it's enterprise scale. Really, we need to scale this up. It cannot be singular labs in separate organizations. We need to have organizational-wide rollout, enterprise scale being pursued to have the impact that we need to have. And this is being pushed further in terms of supply chains. If you're a contract research organization, if you are a CDMO or a contract service organization, these partners will want you to have certification. It benefits them in that they can now report on their supply chain and say, look, our partners are also being sustainable and are so not negatively impacting a supply chain we're obligated to report on. So it's really feeding itself, this movement. And again, the clinical sector too is not to be forgotten about. So I want to take a step back then and see, okay, look, if you're an individual, how can you contribute to this overall goal? So what I would certainly recommend is our ambassador program, a completely free 2-hour program that's online. It's self-paced learning, but really gives the individual power to educate themselves, to see what is involved in being a sustainable scientist. We cover these 4 key areas: energy, waste management, water, and green chemistry. And you become part of a community that will really help each other. If you have labs across the globe that are doing the same thing, why not learn from each other? This is how we're going to move forward, by having a community that helps one another out. And so it's really about practical ideas that you can implement and communicate with your team. Going one step further, then we have an accredited professional program, and this is really to become a green lab expert. If you find yourself already having these conversations in the lab, but maybe don't have the well of knowledge to pull from, this is for you. It's completely self-paced, but it is in-depth knowledge that we're looking at here. And certainly if you want to be an industry leader in sustainable science, this is the first port of call. You'll earn a certificate upon completion, something that we love to see shared on platforms like LinkedIn, and we can, of course, promote as well. And so we focus on 6 areas then in this program, with the inclusion of your procurement, engagement, or bringing the community together as well. And as I mentioned, look, I think the important thing here is that we always look to move forward and take action. So look to bring your lab together. Get in touch with us if you'd like to investigate My Green Lab certification for your lab. Really see what is the extent of your lab operations and where can you make changes. Educate yourselves, engage with the team, create a green lab team. Look at forecasting savings to get further backing from the organization. And ultimately, what we want you to do is spread the word. We need to have that cumulative impact in order to change the culture that is in place and really make it one of sustainability. So thank you very much. Brief, but I hope insightful. A couple of words from me, and I'd be happy to take any questions.
Okay, thank you very much, Mr. Aaron, for the insightful presentation about Green Laboratory. So I think it's very nice presentation because you also share about the success story from some department like in Colorado and also GSK company already get the certification from Green Laboratory. So now maybe before, I think all of participants here also have the question about how to build the Green Laboratory in the company. So now I open for the discussion. We still have some participants in here on site. Please, if you have question, you can raise your hand. From the left side, please. Okay. Mr. Aaron, can you hear my voice? Okay. Thank you.
I can hear your voice, yes.
Nice. Okay, please. Yeah.
Okay, thank you. My name is Dahlia. I'm a student from Open University Indonesia. I'm very glad to hear your presentation, and it's very interesting topic about the green laboratory. My question is, how your laboratory can save more energy? That's the first, and then the second, this is certification. It means we have a journey to achieve the certification. How long is it, and is it any cost for achieve the green energy certification? Thank you very much.
Okay, thank you.
Thank
Mr.
you.
Aaron, clear?
Yes,
Yeah,
yeah,
they have
absolutely.
two questions. Yeah, okay, please.
Yeah, and I'd be happy to answer. So in terms of how do you know, practical steps to reduce your energy usage in the lab, to lower that, it really depends on your specific setting. You know, if I'm talking to a microbiology lab, for example, it's likely that their most impactful step they can make is looking at their biosafety cabinets or fume hoods. That won't be the same case for a lab looking at chemistry, or one that maybe concentrates more on microscopy. What I would say is an important first step in reducing your environment or your energy usage is to first evaluate what is the largest user of energy in your lab. There are very simple things that you can do in many cases with the largest piece of equipment that will make quite significant changes. So if you look at putting in place, you know, a timer, for example, on your outlet, that would ensure that, you know, a piece of equipment could be turned off for 2 hours during during the night. That's going to be massive, rather than having something running 24/7. So I think the important thing really here is to evaluate what you have in place, work with your team and communicate, and there will be very easy options there for you. In terms of how long does the program take, the certification, it's about 6 months on average. It's pretty simple, and directly it's that reason. It's that way for a reason. You know, we understand that you already have a lot of work to be doing in the lab, and so taking on an extra program is something that needs to be, you know, efficient, and really benefit you very quickly. So 6 months. Is there a cost associated with it? Yes, but very slight. You know, from any of the examples that we've just looked at here, labs make back what they ever invest in this program very quickly. And oftentimes, you know, at the organizational level, so at the level of your university, there will be commitments there to be more sustainable. So I would say engage with your university, see if you have a sustainability team, and they will be able to help you in that direction.
Okay, thank you, Mr. Aaron. It's clear, Ms. Dahlia. Okay, thank you. So any more question from the right side or from the yes? Oh.
Sorry for interrupt, Dr. Tepi. There are questions in online.
Okay, maybe you can show in the screen, yeah, in the chat.
Yeah, would you like me to answer the question on the screen? Yeah, fantastic. Yeah.
Okay, you can read in the chat, yeah, please, Mr. Aaron.
Yes. So if laboratory have limited resources, which sustainability initiatives should be prioritized first to achieve the greatest impact? So I'll answer that first. What I would say is first, educate yourself on this. You know, there's a lot of free resources out there. So as I mentioned, you know, look at doing the ambassador program. This will give you a good idea of where in your lab the biggest impact will be. You know, labs are so, they vary so greatly, and will all have different areas of greatest impact. So I would say the first port of call is to investigate the My Green Lab Ambassador Program. Again, it's completely free. It's self-paced, maybe 2 hours worth of content, but will really give you a good idea of the direction that your lab needs to first investigate. In terms of the quickest return in cost savings and environmental benefits, oftentimes it's the largest piece of equipment in the lab that you need to look at. You know, that comes down to an autoclave or a fume hood or a freezer or a fridge. These are the sort of areas that you can make really significant savings with a very simple step. But ultimately, you know, in order to do this, you need to have the full engagement of your team. And so again, I'll just come back to that ambassador program. It's a really great thing to have in place. In my previous organizations, we would have had it as part of our onboarding or our, you know, lab. When you first get started in a new lab, it would be part of the induction. And so it's a great first step to take.
Okay. Thank you, Mr. Aaron. So still any question?
Yes, there are questions in online. Another question.
Yeah.
Yes.
Okay,
Okay.
please.
Yes. So how do you see digital technologies or artificial intelligence supporting Green Lab implementation in the future? Yeah, look, it's something that we're really investigating all the time. Currently, there's already areas that AI is helping people understand, you know, in their organization, where are these, where the change is going to be made, and where can you really make that that impact in cost savings. It will also help you kind of, you know, look further down the line at the impact that might be available, and kind of future-proof the work that you're doing. It's a bit early to tell for us, but it's going to really play quite a large role in the coming years.
Okay, thank you very much, Mr. Aaron. So still have any question? Yeah, from the question 3, please show it. Yeah, this is the question number 3, please, Mr. Aaron.
Yes. So this is a great question, and something I know is quite relevant to you and your teams. Is that, you know, is culture more difficult to change than upgrading infrastructure? I think it really depends on your scenario, but I think that culture is probably more important at this point. What we would say, and what this program really kind of identifies, is that you can have the most sustainable and green infrastructure around your lab and for your teams, but if they don't know how to use it and why they should use it, it won't be sustainable. So, you know, you really can't put the cart in front of the horse here, and so, you know, getting engagement with your lab team, it's already happening. Certainly in the younger generations, there's just more interest to be a more sustainable, to work for a more sustainable operation. Culture is always is a difficult thing to put in place, and it takes time. But there are small steps, making things engaging, letting people see that the change that they can make will help drive that. And you know, having the support there from the organization, you know, is really important. So I would think, look, culture is definitely the one to look at. And in terms of how to be successful in behavioral change, it's support from the institution. You know, give people an outlet for this. You know, if they have interest, create a committee for it. You know, create channels of communication so that if somebody comes across a more sustainable option or a strategy they can put in place in the lab, help them share it. you know, that those channels of communication are really, really valuable to help the whole organization grow and the culture change.
Okay, thank you very much, Mr. Aaron. Still question? Finish.
Yes, there are two questions more.
Oh, more. Okay, please,
Two questions
Mr.
more.
Aaron, still they have two
Yeah.
questions more. Yeah, this is the question number
Okay, so the
4.
Indonesian FDA has developed its own green lab assessment tool. What recommendations would you give to ensure such a tool continues to drive meaningful improvement? Yeah, look, I think in this case, you need to stay relevant and fully understand how sustainability is changing in the world. The important thing here is the continued development and you know, getting the buy-in from labs. I think what can be difficult is the commitment and the workload that can take. That is why I would recommend My Green Lab certification as a simple way of moving forward with it. But making sure to have teams around this, not having it rely on one person's shoulders, will make sure that this lasts much longer than one person's interest. Is this a new question?
Yeah, I think please back to the question number 4. Yeah, okay, now Mr. Aaron.
Yeah, so that's what I just mentioned there.
This is the
is
number
that really
4 before, yeah?
I think we've answered these.
Yeah, this is the number 5.
Yeah.
Yeah, this is before. This is the number 4, yeah? Already, yeah.
Yeah, this is the question I've just answered, yeah.
So this is already answered by Mr. Aaron, and this is the next. Okay, this is the number 6, please, Mr. Aaron.
Okay, fantastic. So yeah, look, the ambassador program is completely free, and just search it on your browser, and you can sign up as an individual. This is, you know, there is no cost associated with it. You can do it in your own time. I would certainly recommend it for any of your team who are interested in looking at sustainable operations. In terms of performance indicators for to measure the tangible impact of ambassador program. It's difficult to tie a direct impact between the ambassador program uptake in an organization and their you know, reduced environmental impact, mostly because we don't have, you know, you won't have the tools in place. But what we can say is that any of the organizations that we find are successful in their My Green Lab certification in achieving real environmental impact have, you know, have a structured program in place to make sure that their individuals are trained to the ambassador program, and are aware and are knowledgeable and have engaged with lab sustainability.
Okay. Yeah, okay, thank you, Mr. Aaron. Please, still any question from participant? So, Mr. Aaron, so I think maybe in the last some few...
I want to ask.
Oh, yeah, yeah, okay. Still any question, please?
Okay.
Please.
Okay, thank you. My name is Rosa. I'm from NQCL DF Indonesian FDA. I want to ask about green laboratory and green building. Many BPOM or Indonesian FDA laboratories operate in existing buildings that were not originally designed with green building principles. In your experience, how much can laboratories improve their sustainability performance without constructing new facilities? Thank you.
Okay, thank you.
Yeah, it's a brilliant question. It's a brilliant question, and it's something that we've worked with a lot. You know, we need to be able to empower teams in any situation to be able to make impacts. You know, and your infrastructure is something that is going to define a lot of that. But what I would say is that there's very simple, in most cases, cheap kind of retrofitting options there that can help you. You know, if you're looking at LED lighting, for example, is a huge one. You know, aerators that you can put on the end of a faucet that will keep the pressure but will reduce the actual amount of water being used are very, very inexpensive. We mentioned earlier outlet timers that can you know, be placed in between your equipment and your energy infrastructure. Again, very, very inexpensive, but can allow you to, you know, switch off a large piece of equipment for multiple hours during the night, and that's going to be huge on your electricity usage. So what we want to do really here is focus on those things that you can achieve. You know, there is no point in it's not going to benefit anyone if we are constrained by looking at, you know, things that we can't do. You know, if your building is older, if your infrastructure is not designed in a sustainable manner, that's okay. Look at something else that you can achieve, because there's no, you know, we're not going to make any difference otherwise. And so, yeah, look, the culture in the team, having your lab set up in a more efficient manner is always going to be beneficial and is always achievable.
Okay, Mbak, it's
Yeah,
okay.
thank you.
Thank you, Mr. Aaron. So maybe before we finalize our discussion, I have the one question for me to Mr. Aaron here. So I think beside the green laboratory, yeah, we also sometimes know about the green building, yeah, because in Indonesia FDA now, of course, we prepare for the green laboratory, yeah. But of course, we have a new building and also we have the old building. Yeah. What is the difference between green laboratory and green building? Maybe you can explain more what is the focus here for the green laboratory and also the green building.
Yeah, I mean, look, I can't speak too much to you know, green building, in that what we look at really here is what we want to concentrate is on the team within the building. You know, I kind of, again, bringing it back to the fact that you can have the greenest building you know, ever designed, but if your team does not know how to use it in a sustainable way, you won't see any of the benefits from that. So what we really want to focus on is the individuals within that building. You know, they will take these learnings and they will move on. They will, you know, come from university, they will go to a new lab. We need those skills to be transferable. Absolutely, green buildings will allow you to be much more energy efficient, but it's really about being able to operate in any of these areas is important.
Okay, thank you, Mr. Aaron. So maybe the last question, yeah. Now we still thinking about to propose to get the green laboratory certification, yeah, from Indonesia FDA. Maybe you can give us, or maybe we can we can find, yeah, the tools, yeah, to do the internal audit, yeah. And we can see we already ready to prepare for propose the certification or not. Maybe you can share with us the tools how to do the internal audit for that.
I'd be happy to share with you some other examples of how organizations are working in that way. Probably can't give you access to the tools ahead of time, but be more than happy to demonstrate them further.
Okay. Okay, thank you very much for your insight presentation, also sharing experience to us for today. Yeah. So maybe we give the applause for the Mr. Aaron. Okay, thank you very much, and I hope we can see you again, yeah, in the next future, yeah, for another presentation and another seminar. Thank you, Mr. Aaron. So,
Thank you very much. My pleasure.
okay, thank you. So maybe I give back the time for MC, please. Thank you very much.
Okay, thank you, Mr. Aaron Desmond, for the excellent presentation, and thank you once again for Bapak Dr. Andes Tepi Usia, Apotheker MPhil, PhD, for expertly moderating the session. So again, as a token of our sincere appreciation for the valuable insight and expertise shared during this session, we will now proceed with awarding of a symbolic certificate to our distinguished speakers. Yeah. Okay, so this certificate is presented to Mr. Aaron Desmond, and ladies and gentlemen, please join me once again in giving them a well-deserved round of applause. And thank you for Mr. Bapak Tepi for moderating this session. Thank you.
Okay, ladies and gentlemen, we have now arrived at the final scientific session of today's seminar. Emerging contaminants and newly identified risks continue to challenge food and drug safety system worldwide. Advances in analytical sciences are therefore essential to ensure effective monitoring, risk assessment, and public health protection. Our next session is entitled Recent Trends and Advances in Food and Drug Contamination Testing, Nanoplastic. This session will be presented by Mr. Jackie, Food and Drug Contamination Testing Specialist. This topic addresses recent scientific developments and innovative approaches in contamination testing, including the growing concern regarding nanoplastic contamination and its potential implication for human health and the environment.
Okay, without further ado, I would like to invite the moderator of this session, Ibu Dr. Anda Maya Gustina Andarini Apotheker MSc, to lead the session. And to our speaker and moderator, the floor is yours.
Thank you for announcer. So very good afternoon for distinguished guests, respected colleagues, and also ladies and gentlemen. So it is my great pleasure to welcome all of you to the very important technical session. This is the last session for today. Hopefully all of you still have a spirit to hear the very important presentation entitled Recent Trends and Advances in Food and Drug Contaminant Testing. So I'm very lucky that our speaker is Indonesian. So we will do campur-campur ya Pak, because actually he is Indonesian, and then just introduction about the contaminant. Today contaminant testing is undergoing remarkable transformation. Analytical laboratories are no longer expected merely to detect conventional contaminant, of course, yeah. No, because instead of they are increasingly challenged to identify emerging contaminants at ultra trace level. This is very important. Respond to rapidly evolving international regulation, of course, and then provide scientifically sound data that support regulatory decision making and public health protection. Jackie will talk about the food and drug contaminant remain one of the most important global safety challenge, of course, and then contaminant such as pesticide residues, veterinary drug residues, mycotoxin, persistent organic pollutants, PFAS, heavy metals, and processing contaminants have become issues that transcend national borders. And we are pleased to have Jackie from Water Corporation. Yeah, Jackie is Food Environment Market Development Manager at Water Corporation based in Singapore. And Jackie drives growth through strategic marketing and sales enablement, market intelligence, lead generation, and competitive positioning to expand adoption of analytical solution across the region. So prior to Waters, Jackie spent more than 12 years in Shimatsu, yeah, in progressive roles spanning application chemist, product specialist, senior product specialist, and also assistant product manager supporting GC, GCMS, and also portfolio across Southeast Asia, South Asia, and India. So Jackie holds BSc in analytical chemistry from the National University of Singapore. It's very famous university, difficult to enter there. Iya itu nggak tahu nomor berapa tuh di dunia ya Nus ini. So
jatuh Bu, itunya udah jatuh sekarang.
Oh ya?
Iya.
Aduh, tapi
masa waktu itu masih tinggi waktu saya masuk di NUS.
Tapi ya itu tetap the number one ya. Di Singapura nomor 1 tetap. Oke. Please Jackie, the time is yours. Silahkan, nanti ada 45 menit ya. Nanti akan diikuti dengan tanya jawab. Silahkan Jackie, terima kasih.
Terima kasih Ibu. Selamat siang Bapak-bapak, Ibu-ibu. Yes. So happy ya, Indonesian representation.
Yeah, Indonesian, but now the citizen is Singaporean.
Iya. Iya sih, paspor Indonesia masih kurang hebat lah ya. Jadi lebih gampang saya pakai paspor Singapura. But that is not the main point here. So the main point today is for me to share with you about the recent trends and advances in the food and drug contaminant testing. So I think Ibu just now mentioned there is a lot of like emerging contaminants. We have heard the word emerging contaminants a lot. And I think this is one of the biggest challenges that we are seeing now in the world of analytical science. So here is this. We are now in the new era. It's the new generation of contamination testing. It's no longer about the known contaminants these days.
The operation of the slide?
Oh, sorry.
Not,
AV team, can you switch the slide, please? No one is seeing my slide. Yeah, I will change the slide from here. All you need to do is to switch the slide to my HDMI cable, please. Extend, yeah. Sorry for the technical difficulties.
Always technical.
Yes. Nggak ada? Oh, okay, great. Oh, thank you, Ibu. I didn't notice. Apologies. Yes. So this was my title. But I think more importantly, we talk about emerging contaminants, yeah? So contaminants are no longer about the known contaminants. Nowadays, we are looking at newer and newer things. Most of the time, it doesn't even have a regulation yet. So these days, we need to be able to, all our laboratories, we need to be able to detect all these persistent chemicals, and we also want to be able to characterize emerging particles. Then, of course, as a very important organization that we have here today, we talk about regulation and how we can respond to evolving regulations. That is also one very important thing for our laboratories to be doing. And so, I'm not sure whether are we blocking this line in the middle? But I will give you the spoiler to the end of my presentation first. Essentially, when it comes to the recent trends and the things that we are going to do, it is about having the integrated workflows that allows us to have greater confidence in safer food and medicines. So to bring you through this, I will go through 3 different chapters. Where the first chapter, we focus on chemical contaminant. The second one will be physical contaminant. And then finally, I'll give you an example of how a contaminant that is recently regulated can be part of the things that we can analyze. So now we will start first with the chemical contaminant. And that will cover PFAS, the PPCP, and EDC. Don't worry, I'll tell you what they are later. So I believe many of us here are knowing the name PFAS, perfluoro alkyl substances. So these are compounds that has a lot of fluorine. And I think the most famous is Teflon, which we are using in our dry fit shirt, our pants, and so on. Actually, PFAS have been very good until we found that they are harmful. And they are now known as the forever chemical because of how inert they are. So let's just compare how dangerous or how long it will last. Now, the half-life of PFAS on average is 7 years. Comparing it to our coffee, I'm sure the morning coffee is no longer effective now. You're feeling a bit sleepy, right? Because the half-life is only 5 hours. So by now, it is not as effective anymore. Unlike the PFAS. If PFAS enters our body, it will stay there for 7 years before it even reaches half. And by bioaccumulation, it will be a very dangerous thing when it starts becoming a high amount in our bodies. And we have been noticing PFAS everywhere. I'm not sure if you have read the news since the past 10 years, but even as far as North Pole, the polar bear also has PFAS in their blood. So we have heard about PFAS being in baby because the mother having breast milk contains PFAS. We also know that PFAS is found in air, in water, in cosmetics. No, I'm not wearing cosmetic. I'm just saying example, cosmetic. So it's everywhere. And it is now a very persistent problem in the world. And so PFAS are compounds which are so useful in the industry. There has been a lot of attempts. Many companies are trying to replace the legacy PFAS. So the older PFAS, those are we known as PFOA, PFOS, for example, already have found their replacements. But the replacements which are shorter, for example, are equally harmful. So now PFAS category is wide. There are like more than 10,000 compounds that are being analyzed and identified. But the amount of regulated PFAS are still quite different from country to country, from region to region. And it also comes to the point where the analysis of PFAS is not so easy. So what we have noticed is that not only just we noticed, actually Waters was helping a lot of the countries' regulatory bodies to develop the methods. So typically, we would start developing using drinking water. Aqua, for example, no brand, sorry. Drinking water. Drinking water is one of the cleanest matrices that we can use to analyze. So a lot of the countries, when the first regulation for PFAS is released, it usually comes with the drinking water first. And so you can see in many countries, these have already been established all around the world. And just within ASEAN, we also have the newly implemented PFAS method in Japan and in Korea. Then usually after the drinking water method is established, then countries will go into different applications and different matrices. For example, for food. So a region, particularly Europe, is focusing a lot on the amount of PFAS that is in food. And they also rejected a lot of food that has been exported to Europe. So a lot of them comes from Asia because we are now factory of the world. We produce a lot of feed and food. And then we sometimes receive this kind of recall for the PFAS that is found in our exports. But across the entire world, it's not just about water and food. There have been a lot of requirements and new methods that is being released every day. Some of them changed into regulation. So for example, we have the PFAS in firefighting foam that is found in the Singapore method, but we also have the PFAS in food packaging material. In fact, I hear that in Korea, there is also PFAS in cosmetic regulation that is being released this year. So there has been a lot of development and requirements for this emerging contaminant. And in terms of an analysis, let's say if you are starting to do PFAS analysis, I would highly recommend you to look at this US EPA 1633A method. So US EPA 1633 method is a multi-lab validated method, and it is developed, checked and validated across multiple labs in different countries. It is applicable to many different types of matrices, including water, food, and even biological samples. So if you are working on PFAS, this would be something that is very useful to start at. And the one that is really used, the instrument that is used would be an LC-MSMS. So LC-MSMS is an instrument that is very advanced, not so cheap, actually, quite expensive. But we need this kind of instrument because the sensitivity and selectivity is required to perform a PFAS analysis. But PFAS analysis is not just about having an MS. Of course, we know having a good MS makes a lot of difference, but sample preparation plays a very big part in any analysis. So when it comes to PFAS, because it is found everywhere. So it is really important for us to be able to perform the contamination management when we are doing sample preparation. So whether it is sample preparation, contamination management, chromatography, mass spectrometry, data analysis, any of these elements cannot be skipped if we want to be able to do a good PFAS analysis. Now, I'm not allowed to talk very commercial things, so I'm not allowed to tell you this, that waters is being mentioned by all the different regulatories in the world. But you can always refer to all these different methods which are already available when it comes to PFAS analysis. And just to show you the kind of challenge PFAS analysis is, in terms of regulation, for example, the EU requires the amount of PFAS that is reported in a seafood matrix to be less than 2 ppb, 2 parts per billion, and some of them even down to 0.2 and 0.5. So only the use of LC-MSMS can allow us to achieve such a low concentration that is supposed to be regulated. And in fact, if we are looking at water, it will be even lower, sometimes to part per quadrillion. So I just never show you. But if you are a lab looking to perform a PFAS analysis, then I would like to highlight the importance of having an instrument that is robust, because by having a robust instrument, we can switch between different matrices if it is necessary. You can analyze PFAS in food and then to water, and when you switch back to food, you don't have to worry that the system is contaminated. So robustness is something to always take note of when you are choosing an instrument. Next, I would like to talk about another emerging contaminant that is called PPCP, pharmaceutical and personal care products. Things which are being used and thrown away, and they will enter the water system, and they will become our environmental contaminants. So this is also one emerging contaminant that has not been regulated. But there is a lot of monitoring. A lot of countries and a lot of regulatory bodies are trying to monitor these compounds, which includes pesticides that is being thrown into the water. So typically to analyze this, of course, since there is no regulation yet, we highly recommend you to get an instrument high-end like LC-MSMS to perform the analysis of PPCP. So just for example, in here, the analysis can go down to concentration as low as 5 nanogram per liter, which is ppb level. So LC-MSMS may be expensive, but you can use it for many applications, which cover all the different emerging contaminants. And of course, I cannot miss the important contaminant that is known as EDC, endocrine disrupting chemicals. When these chemicals come into our body, it affects our hormone system. Maybe the man will become the woman. Maybe we will get cancer. There are so many things that will happen to us. And EDC is dangerous. Dioxin is the most prominent EDC, the most toxic and the most dangerous one. And yet, in the news that I saw last year, the tahu coming from Java is actually high amount of dioxin. Yeah? So this is a very important concern for all Indonesians here. Dioxin is actually a collective term. It is not just the compound. There is no such compound known as dioxin. It is actually a category of compounds known as PCDD and PCDF. And there are so many different combinations of chlorine. It can be 1 chlorine, 2 chlorine, or even 10 chlorine. But there are only 17 of these congeners which are toxic. And when they are toxic, they are really toxic. You know why? When you look at this structure, for example, TCDD is tetrachloro and then the DD lah. It is so planar that it will fix into our DNA. Nah, jadi DNA kita kan sangkut. Waktu mau replication, it cannot replicate. It will break down. And that is why it causes a lot of disruption to our genes and our hormones. So the analysis of dioxin is something that is really concerning. And there are two techniques in general when we are coming to analyzing the dioxin. So a standard method is known as the GCMS-EI. The second method is also GCMS, but it is based on a soft ionization known as APGC. So here I just want to introduce you this soft ionization technique because it actually gives even better results for the dioxin analysis. If you are familiar with the GCMS, so this is usually how EI looks. When we are performing an EI, electron ionization, we break down our molecules to many fragments. So when you have many fragments, the molecular ion is typically low. However, using a soft ionization, we are able to keep the molecular ions intact. And as a result, when we are producing MRM transitions, we can choose a better precursor ion, and the product ion is also higher in intensity. So as a result, the MRM transition is not just sensitive, it is very selective. And this can be very helpful when we are performing analysis of emerging contaminants like dioxin. So good that we can actually go down to around 1 femtogram on column. So of course, the case of Indonesia is dioxin is actually quite high. We don't need an instrument this sensitive. But we also hope to be able to eradicate it, and eventually we want to have the future-proof instrument that can go down to such low concentration levels. So having talked about chemical contaminants, I would like to introduce you some solutions for emerging physical contaminant, normally known as the micro and nanoplastics. So I'm sure you have heard about microplastics because plastics are the same issue as PFAS. They cannot degrade. They will just break down to smaller and smaller and smaller sizes, and they stay in the world. When fish eat them, we eat the fish, microplastic, nanoplastic come into our body. So again, we die. Anyway, we will die. Just we know how we die, okay? I hope we don't die from this. So micro and nanoplastics' main issue is that it has very high surface area to volume ratio. So it tends to trap different contaminants that is chemical because of the physical surface area. And usually we want to be able to find out where these microplastics are from. We want to characterize this microplastic or even nanoplastic. But the challenge is the size is too small. So I think many of us here are familiar with the analysis of microplastic. A pyrolyzer GCMS or a spectroscopy with microscope will allow us to do identification and even visual inspection. But what about nanoplastics? A lot of people still cannot find a solution for nanoplastic. So even though I don't want to be commercial, I have no choice but to show you our product. Because this is the only solution currently I'm aware of that allows us to analyze and identify nanoplastic. So it comes with two parts. The first part is FFF. This looks like an LC, but actually it is force field fractionation. It is a kind of you can think of it as a centrifugation, but at a very micro scale that allows us to do separation. And then the detection is by two different detectors. We have MALS detector, which is multi-angle light scattering, which allows us to do characterization of mass, size, and so on. And there is also a differential RID, refractive index detector that is used in LC. DRID can allow us to do universal detection, concentration, and also DNDC determination. So combination of these detectors and the FFF separation, we can separate out the nanoplastic and then even do identification of these compounds. So how does FFF work? I'll just give you a brief introduction lah, yeah? So field flow fractionation. Imagine we are driving on highway. No need to imagine, this is daily life in Jakarta, yeah? The traffic jam. Imagine you can abandon the car and then you can go away. The car will still move by itself. In a traffic jam situation, everybody will get out of the car and then run away. You will be faster than the car, correct? Now, similar situation in this FFF. We let all the particles flow from left to right. But at the same time, there is an additional flow that is pressing everything so that it is forcing the car to macet like that. Then humans, which are smaller in particle size, will be able to run and go to the front. So in this very simple analogy, I introduce you how flow fractionation can allow smaller size particles to be separated from the bigger particles. So this is one way that we can do the separation of nanoplastics from microplastic. And in addition, the technology that is applied in our product is actually this EAFFFF. Long name, electrical asymmetrical flow, field flow fractionation. Now, in addition to just creating a macet, we actually add electrical field. So all the nanoparticles that are flowing out, if they have a surface charge, they will be separated from similar size particles with different charge. So two charge, for example, will be moving faster than a one charge, which will be moving faster than a neutral particle. So this allows us to do more separation. And when we are doing the detection, we have multi-angle light scattering. So multi-angle light scattering is a very interesting technology that is used a lot in the pharma industry, but it is also very useful for the area of our nanoparticle characterization because of these two principles which are being applied. So if you would like to find out more about these two principles, we can discuss more later, but essentially, the mouse allows us to do true size determination. No need calibration. We will be able to confirm what is the size immediately. We know what is the molecular mass. We will know what is the identity of the nanoparticle. So this is a very powerful characterization tool. And with that, chemical and physical emerging contaminants covered, I would like to go into the third part where I will show you a real-life application where something was emerging but now become regulated compound. And that would be NDSRI for drug. If you're in pharma, you'll be familiar with these names. So maybe around 10 years ago, nitrosamine became a very important concern because nitrosamine was found in a lot of the drugs that is related to diabetes. And so these are known to cause cancer in human. It's very dangerous. But along the way, as nitrosamine is being regulated, we found out that nitrosamine can also react with the API. And when nitrosamine plus API, you get NDSRI. So this is the full name if you are interested, nitrosamine drug substance-related impurities. And so NDSRI can be very hard to identify because it is specific to every API. It always changes, and it can be very hard to find. So when it comes to the limits of nitrosamine, I would like to introduce this term called AI, not artificial intelligence, yeah? This is acceptable intake. So because of the pharmaceutical regulation, the amount of nitrosamine is actually regulated at such AI limits. And AI limits are already very low. For example, for NDMA, it is around 96 nanogram. But dependent on the drug which we are taking, for example, a diabetic person will take the drug every day, right? And for valsartan, the daily consumption is around 300 milligram, 320 milligram. So when we are dividing this AI with the daily dose, the target concentration that we need to analyze is around 0.3 ppm. So this is a very low concentration. If we are looking at, for example, NDMA in API, API usually is very low in volume, so it's easy to analyze. But when we are looking at final products, final drugs, the concentration is much lower. So a method that is supposed to be used for the analysis of such compounds, the nitrosamines, need to be a high-end instruments. And so we know we can use LCMS for this analysis. Now, but then in early 2023, we found out, right, nitrosamine can react with API to form NDSRI. And the AI, an acceptable intake that is being regulated is only 18 nanogram per milliliter. That means it is even lower concentration limit, much harder to analyze. And so here comes the instrument that is future-proof. By having an LCMS, not only is it sustainable, you can see because it has an ACT label, not only is it able to perform analysis of simple nitrosamine, but it also is future-proof enough to work on NDSRI. So using the software, it is able to perform the analysis of 9 nitrosamines in a metformin drug, for example. So this is very good. But at the same time, it is already a sensitive enough instrument that allows us to work on also NDSRI. Even if the API keeps changing, the instrument is sensitive enough, it is future-proof, it allows us to keep track with emerging trends, and we can still meet them. So here comes to my final slide. It is the summary. We know that contamination testing has been expanding beyond known residues. So we need to be able to predict what is the future. But no one can predict the future. So the best way is to have an instrument that is most advanced, selective, for example, that allows us to perform the analysis in future. And emerging particles, even physical testing, will require new characterization strategies. And of course, regulatory response is reshaping the testing priorities. But what doesn't change is that across food and drug safety, right, the analytical challenge is shifting from detecting what is known to workflows of things which might be coming. So we have to create workflows which are selective, sensitive, and also flexible enough to target whatever that may be coming next. And so with that, I would like to thank you for your attention.
Please give applause to Jackie. Jadi menarik sekali ya Jackie sampaikan tadi. Jadi sebenarnya limitnya juga belum internationally sama ya. Untuk
Maaf Ibu, lambat sedikit.
Oh,
pelan-pelan.
Tadi Jackie bilang, nanti kalau ada yang tanya bahasa Indonesia very welcome katanya karena.
Tapi harus pelan-pelan ya Bapak Ibu.
Oke, jadi tadi sudah disampaikan ya masalah kontaminan testing expanding itu ada PFAS ya namanya, PCP dan ADC, kemudian untuk emerging particles need new characterization itu bukan hanya mikroplastik tapi sampai nanoplastik.
Betul, Bu.
Dan itu pernah menjadi isu juga di kosmetik untuk mikroplastik, tapi di pangan pasti lebih serius. Kemudian juga untuk nitrosamin ini juga merupakan isu juga di ASEAN juga sudah di harmonisasi untuk melakukan testing nitrosamin supaya sama. Dan ini kalau saya lihat dari Jackie presentasi tadi, limitnya pun juga ternyata region juga beda-beda ya. Gitu. Oke, silahkan Bapak Ibu kalau ada yang bertanya. Yang dari floor dulu, saya beri kesempatan. Bapak satu ada lagi. Udah Bapak dulu, silahkan Pak.
Thank you very much, Jackie, for your inspiring presentation. I really interesting with the micro and nanoplastic because this is what we facing because in Indonesia, as you know, that our waters mostly now we consume from bottled waters, right? in many countries, they are still using the filtration so that the household can consume directly from the tap water, right? As in Singapore, maybe in some place in Malaysia. But in Indonesia, actually we have the technology, but I don't know why it's not applied. So that the case of the microplastic is really huge and really danger for us, because not only in cosmetic, as moderator said, but also in particularly in our food that we consume every day. My question is, in your presentation regarding the waters instrument, is it possible to measure directly what's the concentration of the plastic inside the sample, or what was the distribution as well as the particle size? Thank you.
Thank you for your question. Yes, so the technology that was shared earlier, FFF mouse with the DRID, it allows us to do the absolute concentration measurement. So of course, a lot of the challenges for micro and nanoplastics still come from the sample preparation. So what I have not shown you is how we can actually get to the stage where the samples can be injected into the FFF. But assuming all the filtration is done, we get to the known amount of sample volume, we can actually do the exact concentration of our nanoparticles after separation by size. So of course, because the detector also have the absolute DNDC calculation and also some other kind of characterization, it is not just for identity, it is also used for quantitation. So it is a 3-in-1, separate, identify, quantify.
Thank you. Is there any other technology that you already apply, because in your presentation you're just focusing on the optical measurement, right?
Yes.
Actually, the other technology is you can use the fluorescent measurement so that you can measure the particle size of the microplastic. Thank you.
Bisa ditunjukkan?
Yes. So sir, would you mind repeating your question again?
Any other technology that you already apply in these devices using the fluorescence detection method? Thank you.
Fluorescence detection.
Because the microplastic, when you add with some reagent that fluoresce, it could be different in the fluorescence, right?
Correct. So fluorescent detector is a detector to a HPLC, which is not used here. In fact, right, in the picture that you see here, even though this is an LC, we are not using LC as LC. We actually only use the pump to maintain the flow, because only with a stable flow, then we can perform this FFF. So actually, there is no column, and therefore we don't use the standard detection like fluorescence or other, even QDA, as in mass detector. So FFF mouse is a very unique combination as itself. It's a standalone. We cannot really combine the standard LC detectors, unfortunately. But the good thing is, because these can do their own detection and also the quantitation, characterization, we actually can do it directly. Whereas in the case of fluorescence, we usually have to do derivatization. And that is not required in this case. This is all absolute measurement, and it is direct. So we can see the answer directly. It is very unique.
Sudah Pak, cukup. Oke, ada
Terima
lagi.
kasih, Pak.
So any more question from the floor? Yes. Oh, okay.
Okay. Sir
Yes.
first, ma'am later, ya.
Oh.
Yeah, my name
Ladies.
Purba. Bahasa Indonesia ya?
Iya, silakan Pak.
Oke.
Tapi pelan-pelan ya.
Oke,
Nanti saya nggak ngerti.
Iya, ini untuk awam.
Nanti Pak, awam itu?
Masyarakat secara keseluruhan, secara general.
Oke, thank you.
Nah, secara general, ini kan akses kepada makanan-makanan yang terkontaminasi itu jadi sangat luas ya.
Iya Pak.
Bagaimana advice dari Pak Jeki untuk masyarakat umum supaya bisa memilah dan memilih yang mungkin lebih sedikit kontaminasinya. Thank you.
Pertanyaan ini mungkin Ibu yang jawab lebih cocok ya.
Tapi
dari Backpom.
Jadi pertanyaan sebetulnya apakah so is it possible to consumer to define that ini kecil kontaminan ini lebih susah juga dengan.
Nah ini ya sebenarnya di Eropa is wait I'm better in English. So this is possible in regions like Europe. Now just now I show you one RASFF is a rapid alert system for food and feed. So in Europe they will do testing, which is why there is a lot of testing services which can perform the check for all your different contaminants and to find out that if the food is safe for consumption, then they will import. And actually, if I remember, I talked to Ibu Cita from BPOM before. She told me that in Indonesia, there is also such a alert system. Yeah, so actually based on the alert system, we can make sure that the food that we are consuming is actually safe, right, Ibu?
Yeah. Sebetulnya di Badan POM kan juga melakukan pengujian. Jadi artinya kita kalau ada kontaminan yang melebihi ada alert system itu warning, public warning. Jadi produk-produk tertentu dia tidak memenuhi syarat. Tetapi bagaimana ya mungkin saya maksudkan Bapak secara bukan kasat mata ya memilih produk kemudian ini kontaminannya lebih sedikit deh. Saya kira kalau seperti itu, itu fresh food atau processed food Pak maksudnya. And so maybe for the processed food you can follow the direction from BPOM, artinya kalau dia punya nomor izin edar berarti oke, kita sudah melakukan evaluasi. Tetapi kalau terjadi sesuatu di produk yang sudah dapat nomor izin edar, pasti kita akan melakukan public warning, recall the product. Jadi konsumen akan tahu bahwa produk ini tidak memenuhi syarat. Tetapi kalau fresh food rather difficult ya, because this is not the responsibility of BPOM of course, and then
gorengan,
jajanan, nah itu yang mungkin agak sulit ya, karena kan kontaminan tidak terlihat.
Kadang-kadang kita pun lihat lah, nah misalnya kalau kita suka makan sate yang bakaran hitam-hitam gitu, polycyclic aromatic hydrocarbon, pH-nya tinggi, tapi kita kan suka, itu rasanya enak. Nah, kalau kita tahu ini bahaya ya jangan makan itu aja lah.
Ya edukasi Pak, kalau yang fresh food mungkin ya. Gimana Pak, sih bisa menjawab atau soalnya kalau gimana konsumen bisa memilih ini yang sehat ini yang tidak. Kalau proses food mungkin yang ada Badan POM-nya lebih mudah ya, tapi kalau yang fresh itu ya cari yang bersih-bersih. Kalau nanoplastik gimana ya?
Plastik masih belum bisa,
nggak
Bu.
bisa, ya
BPOM harus beli ini dulu.
Bu, beli Bu. Okay, the next question.
Tadi ada Ibu.
Silakan.
Yeah, thank you. I'm interested in nitrosamine analysis, especially in metformin, which has a very high maximum daily dose. We know when we try to align with the LOQ, AI limit, and the maximum daily dose, we will find that we need to prepare a very high amount of sample to prepare, and we find it very challenging. And I want to ask if you have any recommendation or tips or tricks for us to prepare the kind of sample that has a very high amount of maximum daily dose. Thank you.
So I actually was using GCMS mostly. You can sit first. Bu. So I was actually working on GCMS before I started working on LCMS. So I think one of the main challenges for metformin is that when you inject into GC, the temperature is very high. It will react and then it will change its form into NDMA kan. Nah, so the solution is to use LCMS. Because LCMS is room temperature, so it actually helps to solve the problem already. But is that answering your question?
Yeah, we try to use LCMS, but that's the challenge because we need to prepare the liquid sample. That when we extract the very high amount of sample, it's kind of maybe 4 tablets. 4 tablets in 5 milliliters. It's quite challenging, and sometimes we find a crystal in the vial after we prepare it.
I see, I see. I may not be the best person to answer this question for you, so if it's possible, please stay behind because I have my application colleagues who are around, which we can do discussion to talk more about this.
Okay, thank you.
Thank you, Ibu.
Nanti bisa komunikasi dengan yang orang yang tepat ya.
Teleponnya nanti ya.
Oke, masih ada yang di offline? Sudah nggak ada. Nih kita pergi dulu ke online. Silahkan tayangkan. So please show the online. Nggak ada ya? Kok nggak keluar? So still is a technical problem. Technical is always problem.
No problem.
Okay. So this is the question, Jackie.
Okay, so I will just read the first question. Which emerging contaminant do you believe will have the greatest regulatory impact over the next 5 years? This is a very easy question for me because I have been looking at the APEC, Asia Pacific regulation, as well as the global, and I can tell you the answer is PFAS. PFAS regulation is changing so fast. I have never seen any regulation changing within every year. So it is now known to be one of the most dangerous things because it has been accumulating for a long time. And now then we realize the problem. So we are working backwards to try to remove the source of PFAS, but it's not possible actually. Every replacement for PFAS is still dangerous to us. So this has becoming the biggest challenge in the recent years of regulation. So it's really important for BPOM actually to really consider having this method, I would believe.
Yeah, so still PFAS will be next struggling.
Correct.
For face the problem. Okay. Yes, for the second,
Thank you.
how should
Second question.
national regulatory laboratories prioritize investment when analytical demands continue to increase? Maybe.
This is a very big question. I may not be the best person to answer this. I think our mentoree is what? Minister maybe are more suitable to answer this question.
Brand from your minister?
But of course, from my own perspective, I was a chemist. So to me, I believe that having a lab that is capable of doing not just what is currently required, but also what is required in the next 5 years will be the most important thing. So if I can give you an example, one of the companies just this year, beginning of this year, very, very famous brand, selling the baby infant formula. The baby powder, inside there is some toxin, cereulite toxin. Like that, might as well I tell you the brand Nestle lah. They actually voluntarily recall their product because they found cereulite toxin in their drugs. In their baby powder, in the infant formula. So this is an example where if your lab is not capable of doing the testing, you will not be able to react immediately when something like that happens. So I talk to other infant formula companies, like, how come you don't have this issue? And they are like, oh, because we have new instruments. So we are fast enough to make sure that it doesn't happen. Maybe the lab in Nestle that is working on it happened to have older instruments, so they were not able to keep up with the requirements. That's why that happened. And so having that future proofing, at least for the next 3 to 5 years, to me, is one of the most important things for a laboratory, especially when it comes to national level.
Jadi
personal opinion ya.
National Laboratory tidak boleh ketinggalan ya. Harus selalu update the instruments to face the issue.
Iya, Bu.
Nowadays. Jadi begitu prioritasnya ya tergantung nanti apa masalah yang akan dihadapi dan tidak boleh kalah, tidak boleh lebih ini daripada private company.
Ya, kalau sampai masuk berita itu kan malu nanti.
Iya. Tapi jangan keras-keras. Oke, ada lagi?
Ada. There are 6 questions in total actually.
6.
Oh, 6. Oke. What are the biggest analytical challenges to PFAS determination across different metrics?
This answer is contamination from environment, in short. Because PFAS is everywhere. Like literally when we go into the laboratory, some of our colleagues, if we wear makeup, even the eyebrow shadow, for example, you will create contamination. Our vial is having the PTFE cap. Our pipette tip is made of plastic. Everything is a source of contamination. And so contamination management is the biggest challenge in PFAS determination. If you want to find out how, talk to me afterwards lah.
Further.
If not, it will become commercial already.
Further discussion and further business.
Yes.
Okay, the next, is it still? How do LCMS MS and higher resolution MS complements each other in modern contaminant testing?
This is a wonderful question. LCMS MS is normally known as TQ, which is a routine instrument. High resolution MS, for example, QTOF, or some technology like Orbitrap, or even like cyclic IMS, all these instruments, right? These are high resolution, which can give you accurate mass. And typically, high resolution MS would be useful for identification, especially for unknown. So this has been when something is being discovered by a HRMS, it will become routine, which will be analyzed by a TQ, LCMS-MS. So they actually complement each other significantly in the sense where HRMS will do the discovery, and then LCMS-MS will do the routine job. That is the current situation in a lot of the analysis lah. But we can actually also see more and more labs are investing into getting HRMS, because that is the fastest way to get into future proofing when you want to be able to analyze what may be coming in the next 3 to 5 years. HRMS will always give you the answer.
Okay, so LCMS for routine and high resolution MS is for special case.
Discovery, identification of unknown, for example.
Okay. Yes, because I also heard that the participant from Zoom is occupied until 1,000 ya Bu Mimin. Ada 1,000 partisipan di Zoom. Jadi pertanyaannya banyak.
6 questions is quite low for 1,000 people.
Yeah, of course.
But it's okay, we can stop for now.
No need so many questions. Okay, next nggak apa, satu lagi ya.
Okay.
Boleh? Soalnya pas nanti. Okay, so what analytical challenge remain for microplastic and nanoplastic characterization?
So this question was partly answered earlier. I think the biggest challenge for micro and nanoplastic is the separation, the sample preparation. Getting them out can be very tough in the first place, because filter paper, for example, the filter paper we use in the lab is too big for microplastic, let alone nanoplastic. So we really need to find very, very fine pore size in order to separate. But that will mean it's very slow. So currently the biggest roadblock for this analysis is actually the sample preparation. And how close are the current analytical technologies to routine regulatory testing of nanoplastics? Actually, if you're already working on microplastic, you already have the sample preparation tool, then nanoplastic is not a problem. Then you only need to have FFF mouse and a way to do contamination management. Similar to the case of PFAS, nanoplastic is also found everywhere. Like Ibu just now mentioned, even cosmetic also will have, right? So contamination management is a similar problem that we have with PFAS. And of course, as long as you can do the sample preparation, it will actually be something that we can already start implementing routinely.
Kalau Bu Mimin bisa ya, nanoplastik udah bisa ya? Oh, udah bisa. Di lab kita sudah bisa juga untuk nanoplastik. Oh, very scare about the nanoplastic.
Yes.
Yeah, okay. So ada satu lagi. So still one more. what the lesson have regulators learned from the global nitrosamin issue?
This question I think is for a regulator, Ibu.
Ini gimana Bu? Artinya karena memang nitrosamin ini banyak ya, selain obat, kosmetik pun juga ada yang nitrosamin. Jadi untuk ASEAN sendiri sudah diharmonisasikan, artinya ya ini memang isu-isu yang cukup besar, bukan cukup besar, sorry, lebar artinya di produk apapun. Ya, jadi what's lesson have regulators. Ya, oke. Saya kira itu cukup ya. Terima kasih Jackie dengan full insight of your presentation tadi, cukup menambah wawasan kami, dan mudah-mudahan next kita bisa tetap untuk diskusi ya, for example untuk more issues of the contaminant, entah di pharmaceutical, food and cosmetic, health supplement, etc.
Yes, terima kasih.
Thank you very much.
Bu.
Berikan applause sekali lagi untuk
Thank
Jackie.
you everyone.
Thank you very much, Mr. Jackie, for excellent presentation, and thank you for Ibu Dr. Maya Agustina Andarini, Apoteker MSc, for expertly moderating the session. As a token of our sincere appreciation for the valuable insights and expertise shared during this session, we will now proceed with awarding of a commemorative plaque and certificate to our distinguished speaker. May we respectfully invite Ibu Mimin Jiwawinanti, SSI Apoteker, to come forward and present the plaque to Mr. Jackie. And we invite Ibu Maya Agustina to present the certificate to Mr. Jackie. Okay, ladies and gentlemen, please join me in giving them a well-deserved round of applause.
Okay, ladies and gentlemen, as we conclude the first day of our seminar, we are reminded that innovation and sustainability are not separate ambitions. Together, they constitute the foundation of future laboratories that protect public health and contribute to global well-being.
Ladies and gentlemen, we have now reached one of the most anticipated moments of today's seminar. But before we close this session, it is time for our lucky door prize draw. And now we would like to invite our social media team to deliver this session. The floor is yours.
Assalamualaikum warahmatullahi wabarakatuh. Selamat sore Bapak Ibu peserta seminar kita pada sore hari ini. Terima kasih tetap bersama kami hingga sore hari ini. Untuk kegiatan ice breaking kita, tepatnya pembagian door prize, kami akan melanjutkan dalam sesi bahasa Indonesia. Dan ini akan berlaku juga untuk peserta yang hadir secara daring. Jangan lupa untuk standby di depan laptopnya karena kita akan memutar wheels tentunya. Bagaimana selanjutnya Kak Ici?
Mungkin sebelum memutar wheels kita akan ada pertanyaan dulu kali ya.
Oh iya, untuk pertanyaan itu kita untuk Bapak Ibu yang tetap standby bersama kita di yang secara luring ya. Jadi untuk itu kita akan ada 2 pertanyaan, tentunya akan ada hadiah menarik yang telah kita siapkan. Pasti Bapak Ibu jika telah diberikan pertanyaan bisa langsung angkat tangan dan memberikan jawaban terbaiknya.
Kita langsung aja.
Oke, boleh. Kita langsung saja. Sudah siap Bapak Ibu?
Udah siap? Oh ya, ada titipan pesan untuk nanti untuk pemenang atau peserta yang berhasil menjawab, syarat dapat hadiahnya harus follow IG P3OMN dulu.
Ya, Instagram P3OMN dengan username ppomn.bepom. Jadi nanti kita cek dulu ya bagi Bapak Ibu yang sudah angkat tangan dan dinyatakan benar oleh panitia, maka nanti bisa menunjukkan bukti telah mengikuti Instagram ppomn.bepom. Mungkin bisa kita kasih dulu waktu sekitar 1 menit untuk bisa membuka akun Instagram masing-masing dan melakukan follow di akun ppomn.bepom. Silahkan Bapak Ibu.
Di akun Instagram p3omn.bepom ini tentunya Bapak Ibu akan mendapatkan informasi yang sangat bermanfaat dan juga menarik yang terus akan diupdate oleh tim sosial media dari P3OMN. Jadi mari kita follow Instagram P3OMN.bepom dan dapatkan informasi yang menarik dan kadang juga ada kuis juga loh Bapak Ibu.
Kita selalu ada kuis tiap bulan dan tentunya kuis kita berhadiah Bapak Ibu. Jadi silakan jika mengetahui jawabannya bisa langsung di komentar, tinggalkan komentarnya di kolom komen, nanti akan dihubungi oleh tim P3OMN untuk memberikan hadiahnya jika jawabannya benar. Oke Bapak Ibu, apakah sudah follow akun Instagram kita? Sudah ya, kita langsung saja ke pertanyaan pertama ya Bapak Ibu. Silakan pertanyaan pertama.
Apa judul seminar hari ini?
Oke, sudah.
Oh, gimana? Maaf.
Oke, 1, 2, 3 ya Bapak Ibu, 1, 2, 3. Tepat pada hitungan ketiga langsung angkat tangan.
Panitia udah siap belum? Udah?
Panitia udah siap belum? Oke, 1, 2, 3.
Yang mana?
Bapak yang, yang baju hijau. Ya.
Ini pertanyaannya apa, Kak? Judulnya ya?
Iya, judulnya.
Judulnya International Seminar Perspective from the Five Continent dengan subtemanya The Laboratory's Futures dengan temanya Sustainability in Innovations and Operations.
Oke, bagaimana Bapak Ibu, apakah benar?
Benar?
Betul ya? Betul.
Yang pertama, jawabannya gimana?
Benar?
Benar, jawabannya benar. Yang pertama mungkin kita bisa tahu dulu Bapak atas nama siapa dan dari mana?
Perkenalkan, nama saya Okta, saya dari perusahaan bidang manufaktur, tepatnya di AS Mesin.
Oke, kami mohon izin boleh ngecek apakah sudah follow Instagram P3OMN. Agak ketat ya Bapak, mohon maaf.
Oh boleh.
Meski sudah sore kita tetap mengikuti aturan.
Kebetulan juga sudah Bepom sih Bu.
Oke, emang keren banget sih.
Udah lama. Sudah.
Oke, hadiah kita berikan ya Bapak Ibu, silakan diberikan tepuk tangannya.
Kemudian kita akan lanjut pertanyaan kedua, apa?
Oke, untuk pertanyaan kedua, sebutkan salah satu judul dan narasumber pada materi kita hari ini. Kita 1, 2, 3 lagi ya Bapak Ibu. 1, 2, 3. Ibu yang di tengah. Ya, Ibu yang berkerudung coklat. Silakan Ibu dikenalkan dulu nama dan
Saya Ayun dari Fakultas Farmasi Universitas Indonesia. Tadi session yang pertama itu tentang HPTLC oleh Dr. Tiandu.
Oke, bagaimana Bapak Ibu, apakah benar? Oke, bisa diterima ya. Selanjutnya kita cek berarti ya Instagramnya. Mohon izin ya Ibu. Oke, untuk peserta luring kita sudah mendapatkan 2 pemenang pada hari ini. Untuk hadiahnya nanti akan dihubungi panitia, tentunya akan menerima hadiah menarik dari kami. Selanjutnya untuk Bapak Ibu peserta daring yang masih tetap bersama kita hingga sore hari ini, Silahkan ditampilkan Bapak Ibu Panitia, kita telah menyiapkan wheels sesuai dengan daftar hadir yang telah diisikan dari pagi tadi. Bagi Bapak Ibu yang beruntung tentu saja akan mendapatkan hadiah yang menarik juga. Boleh panitia? Wah, sangat padat, rapat. Bagaimana kita putar wheelsnya ya Bapak Ibu, langsung saja. Kita putar wheelsnya. Selamat kepada Bapak Ronald Hatoguan Manik, apakah masih ada di Zoom? Halo, apakah Bapak Ronald masih bergabung bersama kami di Zoom? Mungkin boleh open mic terlebih dahulu. Kita hitung sampai 5 ya, jika tidak maka kita anulir ke pemenang selanjutnya. 1,
2, 3, 4, 5.
Oke, kita putar lagi. Masih ada kesempatan bagi yang belum keluar namanya. Silakan diputar wheelsnya.
Selamat kepada Ibu Rini Susanti, apakah masih bergabung bersama kita di Zoom? Dipersilahkan untuk open mic.
Halo Ibu Rini.
Halo Ibu Rini.
Halo Ibu Rini. Halo Ibu Rini. Kita hitung lagi sampai 5 Bapak Ibu. 1, 2, 3, 4, 5. Oke, kita anulir lagi. Kita putar lagi. Selamat kepada Ibu Dinda Aulia Fuadi, apakah bergabung bersama kami di Zoom? Halo Ibu Dinda. Oke, kita ambil lagi 1, 2, 3, 4, 5. Tidak ada. Sepertinya karena sudah mendekati jam pulang ya Bapak Ibu, mungkin sudah siap-siap pulang atau mungkin sudah melaksanakan sholat asar. Jadi bagaimana? Kita putar 1 yang terakhir. Jika tidak ada maka akan ada 2 pertanyaan lagi untuk Bapak Ibu peserta luring. Oke, terakhir. Oh, selamat kepada Bapak Muhammad Yamin Anggriaman, apakah bergabung bersama kami? Oh, sayang sekali belum ada yang beruntung dari peserta daring pada hari ini. Mungkin besok kali ya peserta daring. Oke, berarti kita remove lagi ini. Kita langsung ke peserta luring saja untuk pertanyaan selanjutnya.
Panitia sudah siap?
Oke, untuk pertanyaannya mungkin kita langsung saja ya. Oke, untuk pertanyaan tambahan peserta luring silahkan.
Sebutkan nama beserta gelar Kepala Badan POM RI.
Langsung.
Harus benar ya Pak?
Saya John Parlo dari UMKM. Profesor Taruna Muhammad Taruna Ikrar kalau nggak salah ya.
Kita hitung gimana?
Taruna Ikrar, iya.
Oke, kita hitung lagi. Ternyata belum benar ya Bapak, mohon maaf.
Oke, kita minta yang benar ya Bapak Ibu. Kita hitung dulu, tangannya minta tolong izin diturunkan terlebih dahulu. Dalam hitungan ketiga silahkan angkat tangannya. 1, 2, 3.
Oke, kata panitia ulangi 1 kali lagi. Maaf. Siap? 1, 2, 3. Baik, Ibu Ibu yang pakai baju biru dan berpakaian.
Ayo silakan Ibu, perkenalkan dulu namanya Ibu, mohon maaf.
Perkenalkan, nama saya Titin dari UMKM. Jawabannya Profesor Dr. Taruna Ika M. Biomed PhD.
Bagaimana Bapak Ibu? Mohon maaf Ibu masih belum benar. Kita izin sekali lagi ya dalam hitungan ketiga. 1, 2, dalam hitungan ketiga Bapak Ibu.
Boleh ulang lagi.
Tahan, mohon bersabar. 1, 2, 3. Ibu yang berkerudung hijau di tengah, mohon maaf, silakan Bu. Silakan Ibu perkenalkan diri terlebih dahulu.
Nama saya Lina dari Tomang.
Oh iya.
Jawaban saya adalah Profesor Dr. Taruna Ikrar Muhammad Biomed MD PhD.
Bagaimana Bapak Ibu? Masih harus kita anulir ya ke peserta lainnya. Oke, kita temukan sampai dapat yang benar tepat ya Bapak Ibu. Makin sore ini makin diasah nih. Untuk yang salah sudah tidak bisa angkat tangan lagi ya, mohon izin Bapak Ibu. Oke, yang semangat ya, yang semangat. 1, 2, 3. Ibu yang blazer hitam.
Profesor Dr. Taruna Ikrar, M. Biomed, PhD.
Salah padahal sedikit lagi ya Ibu. Aduh sayang sekali. Ya Bapak Ibu yang belum angkat tangan. Oh oke. Ibu yang kerudung biru. Silakannya di tengah Ibu.
Nama saya Sri Retno dari Kementerian Perindustrian. Untuk gelar dan juga nama dari Bapak Kepala Badan POM itu Profesor Dr. Taruna Ikrar Magister Biomed atau MD Biomed PhD.
Oke, tepuk tangan di sebelah sana apakah mewakili bahwa ini jawabannya benar? Setuju benar ya Bapak Ibu, akhirnya pertanyaan satu ini menemukan pemenangnya. Kita berikan tepuk tangan yang sangat luar biasa dulu kepada Ibu. Oke, terima kasih. Yang kurang benar. Oke, sudah benar Ibu. Untuk Bapak Ibu peserta daring, jangan bersedih hati dulu. Karena besok kita masih ada kegiatan seminar kita, masih akan berlanjut seharian lagi besok. Untuk itu kami persilahkan Bapak Ibu untuk dapat bergabung bersama kami mengikuti kegiatan dari pagi hingga sore hari besok. Dan tentu saja juga akan ada kuis lagi untuk khusus peserta daring. Untuk itu kami mohon Bapak Ibu untuk tetap setia bersama kami dan join lagi besok. Terima kasih.
Mungkin sekian untuk sesi ice breaking hari ini. Saya Rosa.
Dan saya Aziza.
Kami mengucapkan terima kasih atas partisipasi Bapak Ibu dan kami mohon maaf apabila ada kesalahan. Wassalamualaikum warahmatullahi wabarakatuh. Kami kembalikan kepada MC. Terima kasih.
Okay, thank you very much for our social media team for delivering the quiz and the door prize in the end of the session. And ladies and gentlemen, we would like to say congratulations to our winner of this session. And thank you for your active participation of today's seminar. And we look forward to welcoming you again tomorrow morning online for the external BPOM and offline at 10 a.m. for the internal BPOM in the Aula PPP OMN.
Okay, once again, thank you very much for your kind of participation for today. Have a pleasant evening, and thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
See you tomorrow. Thank you very much.
Cek. Baik, untuk Bapak dan Ibu panitia seminar internasional dipersilakan untuk masuk ke dalam auditorium karena akan dilaksanakan foto bersama terlebih dahulu.
Tak banyak yang kulihat, foto kita blue, tapi banyak yang aku ingat. Foto kita blue, kita pernah begitu dekat. Foto kita blue, kini memori